Let's talk about muscular physiology. I've been led to believe you guys have at least covered some of the basic anatomy stuff already. We're not going to get super in depth on that. We'll talk about just briefly a few things and then really get into the meat of the topic. Okay. Anyway, all right, so here we're going to talk about the muscular physiology for section 4. This first section, this will probably be most, or if not all, of the 4 hours today, and then I'll cover kind of more general issues in the nervous system. We'll talk a lot about like neurotransmitters and things like that, and then we'll get into more detailed specifics on the CNS, and then talk about the autonomic nervous system after that. Okay, we'll talk about the ANS, but then we already talked about the GI tract, right? I said ANS really fast here. Okay, anyway, so today, muscular physiology. And so we'll talk a little about the skeletal muscles. We'll talk about the mechanisms of contraction. Talk about kind of the controls of that, whether kind of multiple number of levels where contraction of muscles occurs. And then we'll also actually talk about smooth muscle at the end here as well. Just because we've kind of been alluding to smooth muscle control kind of all throughout the course with things like, the GI tract and whatnot. But we'll kind of talk more about that a little bit in detail. So anyway, skeletal muscles. As you might imagine, they connect to your skeleton, right? This is a good thing. Generally, you're going to find that both ends of the muscle are going to be attached to the bone by the tendons, as you guys are already familiar with. And so when the muscle contracts, it's going to be shortening. We'll talk about that mechanism and how that actually occurs. Basically, as you're putting tension on the tendons, it's going to connect, as it's connected to the bone, that's going to cause the movement to actually occur there. And so, as you guys know, the bone that moves that's attached there is going to be called the muscle insertion versus where the originating point will be called the origin there. I also know there's going to be some agonist and antagonistic movement here between muscles, which is good to have that balance there. Otherwise, if you don't, then things can get a little wonky. So different movements depend on that joint and how the muscles end up being attached there. So we saw the flexor muscles typically decrease the angle between two bones at a joint versus the extensor muscles, which should be increasing that angle there. And then we'll see that the muscle that's responsible for movement in a given direction will be the agonist, right? So if I'm trying to shorten the distance or shorten the angle between my humerus and the arm bones, you're going to be seeing that's going to be the main agonist there. So the biceps would be the main agonist versus the antagonist at that point would be which muscle? Yeah, the triceps, right? So you're going to have kind of an antagonistic kind of activity there. So, right, so we'll find where the different mechanisms occur and the kind of reflexes that happen to allow that when you want to contract the biceps, they're going to have a commensurate relaxation of the triceps, right? Otherwise, you'd end up having no movement that can occur there. And so we'll talk about a few different disease states, but one of the problems you run into with certain antipsychotic drugs is a process called neuroleptic malignant syndrome. You guys ever heard of that? Well, basically, it's a rare condition, but you end up getting what we call lead pipe rigidity, and that can be where you basically have kind of inappropriate contraction of kind of both sides of the muscle. So the patient gets very, very stiff. It's very, very difficult for them to relax any of those muscles on either end of that. So you can see that they're both acting as agonists in that case. You're not really going to get any movement that can occur there. So we'll look at the processes that allow for this to happen appropriately when either subconsciously or when you're actually controlling it consciously. So we'll talk a little bit about these in detail. I'm sure you guys have already covered most of these in anatomy, correct? Kind of the different movements you can actually have there. So we'll talk about this more in detail in just a second, but just briefly. So obviously, flexion we mentioned is going to be decreasing the angle here versus extension is going to be increasing the angle. So pretty straightforward. You'll see the abductors and the adductors. I just remember the abductors as being like if I was abducting your leg from you, I would be taking it away from you, right? So, so you can think about this as the beginning of the movie Taken, and this would be like the end of the movie Taken, right? Where you get the daughter back at the end, so you're adducting back. So I always had a hard time remembering like if you go to the gym, you have like the machine where you can like kind of swing your legs open and out. Okay, which one's the adductor versus the abductor? And so that's what I remember is if I'm taking your leg away from you, that would be abduction there. We have the levators, which you can see is going to be kind of raising muscles up there, and then depressors, which will be moving it down. And then the rotator muscles, so either have internal or external rotation there. So again, if you're having like internal rotation, you'll be kind of moving in versus out. So again, pretty straightforward there. We talked a lot about our sphincter muscles already, which are mainly just going to be kind of constricting around an opening that allows for impediment of flow. Either relaxation allows for flow to occur, otherwise you should be kind of squeezing down on there. Okay. We see that a lot of these are going to be under more subconscious control in a lot of cases, depending on the situation, and then obviously we know which ones are going to be under conscious control. So we talk about things like the bladder, talk about things like the rectum, things like that, where you can find some conscious and subconscious control. Okay, the thing I'm more focused on is going to be the skeletal muscle actual function, how we cause those contractions to occur. Okay, so looking at the actual structure of the skeletal muscles, we're going to be seeing that there's going to be a lot of fibers that are going to be running the length of the muscles here. So if we're kind of looking at the bicep, if we were to take a cross section of that, you can see basically where the tendons are going to be attaching to the bone here, and then we'll look at the individual muscle fibers. Do you guys know what these are right here? Actually, the nucleus of the cell there, right? So you're going to find that within each individual muscle fiber, there's going to be multiple nucleuses. There's going to be a lot of communication across the cells here, and you're going to find that they're going to be made up of these kind of individual myofibrils. So you can kind of think of this kind of whole thing here as the cell, multiple nuclei, a lot of mitochondria are going to be involved here because we'll see the energy utilization is very, very important. And then they're going to be made up of these kind of individual myofibrils. Okay, so those myofibrils are made up of several different bands here. And so we'll see that how those function together in order to cause actual contraction or relaxation to occur. So we'll get into much more detail on this in just a few minutes. But again, the plasma membranes here are going to be called the sarcolemma. So you can see this would be the sarcolemma kind of encapsulating this whole thing with all the myofibrils. And at the end of the muscles, they should be basically fusing with the tendon fibers to cause either the insertion or the origin of the muscles there. All right, so we call skeletal muscle, we also call it what type of muscle? Striated, right? So why is it striated? So these nice bands. If you were to look at it under a microscope, you can kind of see these different gradation of colors. I don't think I include any microscopic pictures, but certainly if you look that up, if you Google it, you will see there's lots of different banding that occurs there. So it kind of has a striated appearance. A lot of it has to do with these bands and kind of what makes up the individual bands. So looking at that, we see the eye bands are going to be these light bands, right? So you can see the eye band is right here, kind of this light area. You're going to notice that it really is only going to contain the actin filaments. We talk about muscle contraction, we'll talk about myosin and actin. This is mainly going to contain the the actin filaments. And this section right here is going to be called the Z disc. This is where the actin is actually bound to, right? So it stays connected to the Z disc. And you can kind of think about we'll talk about the sarcolemma, and we'll talk about that being kind of this unit between Z disc here, right? Because this is going to be the contractile unit that's going to actually come together and cause a shortening of the muscle there, right? So anyway, so we have the Z disc holding the actin filaments. You're going to find the A bands are going to be these dark bands. So you can see the A band kind of extending here. And so we mentioned if the I bands are just containing the actin, then you would assume the dark bands contain the myosin, right? So these are going to be kind of larger chain molecules. You can see here, they'll kind of give it a darker appearance. Basically, the H zone. Talk about the H zone being the area where there's really no overlap of the actin and the myosin. You're going to find that overlap is very important to cause contraction to occur, but you can consider this H zone here where there's really no overlap between the actin and the myosin. Here you can see in this picture the myosin versus the actin filaments. Here would be your Z disc, right? And so you want to have some overlap, and we would consider those areas still part of the A band at that point, right? Because they still have that kind of darker color to them than you would see with the just in the I band specifically. So anyway, and again, Z discs are very, very important because this is going to be where the actin's binding, and then when you have a contraction actually occur, you're going to see shortening here. So it should kind of shorten up and cause the actual shortening of the muscle and contraction that occurs there. Have you guys ever covered any of this stuff before? Just all, yeah, okay. So we'll get in even more detail in just a minute. So again, this is just the whetting your appetite, so to speak. So again, that sarcomere is going to be the area between Z discs that ends up getting shortened. There's also this titin molecule. It's kind of a springy protein. Kind of see the titin here. This kind of elastic protein, and they basically are working to kind of hold the actin and myosin in place, kind of help things to stay coordinated with one another. Also, when you see contraction that can occur here, these will end up getting shortened, and they'll kind of provide some elastic force, so that when you cause relaxation, you should see the muscle kind of lengthen back out, right? So when you have kind of relaxation input, it should cause that to spring back out. Okay. Looking at the sarcoplasm itself, so instead of having like cytoplasm in a normal cell, we'll have sarcoplasm. You're going to find this is this fluid between the myofibrils, and so this is going to contain high amounts of things like potassium, magnesium, phosphates can be really important here. Why do you think phosphates going to be really important to have there? Yeah, for energy production, right? So we'll talk about things like phosphocreatine and also ATP, but we need a phosphate there in order for the muscles to develop energy for it to function. Also, lots of mitochondria. Again, very important for production of ATP. So we'll talk about energy requirements and energy utilization in the muscles just a little bit later. All right, so then we have the sarcoplasmic reticulum. This is going to be very, very important because what gets stored in the sarcoplasmic reticulum? Lots of calcium, right? So this is going to be useful for releasing calcium, for reuptaking calcium. So typically in the sarcoplasm, you don't find a lot of calcium that's actually there. If the concentrations are very low because most of it's being held in sarcoplasmic reticulum there. So that'll be very important. Okay, we'll also talk a little bit about motor units. Basically, this is going to be, you'll find that muscle fibers tend to only be innervated by one particular neuron, right? And so you can have multiple fibers that are all being innervated by the same neuron, and that would be considered to be the motor unit, okay? So, a single motor unit, all the muscle fibers it innervates, and basically this allows for those, when an action potential comes along, allows for contraction of all those fibers at the same point, right? And so you can find that you can have very fine control of this, or you can kind of very not so fine control, right? So you're looking at things like, the number of fibers that get innervated by this particular neuron when you're looking at the motor unit. So about 2 to 3 fibers for something like the laryngeal muscles, right? So why is that important to have very fine control there? So I can talk to you guys, right? So I can project my voice, or I can change the tone or intonation or whatever I need to change in order to communicate that. So I can have very, very fine control over that. And you might only find, say, 2 to 3 fibers or so being innervated by a single neuron at that point. Versus, if it's something where I don't need a ton of fine control. So think about things like the soleus. Do you guys know what the soleus does? Yeah, it kind of helps to increase the angle of the ankle essentially, but it's very important for doing what? Walking, standing, right? So if I didn't have any kind of soleus activity, I'd probably be on the floor right now, right? Which is probably where I want to be taking a nap, but that's neither here nor there. But I don't really need a lot of fine control of the soleus, and so you'll find that several hundred fibers may be innervated by just a single neuron. So those motor units would have less fine control, but again, in a lot of cases, you may not really need that, and then that's okay. We'll also find there's this interdigitation. Do you guys know what this means? There's a lot of overlap between the different motor units that occur there. So you imagine that you don't find just like one particular, so for instance, if you had an action potential that came along from the spinal cord, again, notice this is coming from the kind of the anterior horn of the spinal cord. You're going to see that you don't necessarily have just this half of the muscle contracting versus this half. That wouldn't really make a lot of sense. It wouldn't be very useful to us. So there's literally kind of a lot of overlapping of those motor units. So that way you get a much better kind of consistent contraction of the muscle versus just having individual sections binding. It wouldn't really be all that efficient for our needs. So the interdigitation there is useful because then the separate motor units can help one another out essentially. And you'll find that as we start to have communication, we start to have those action potentials, that the smaller motor units tend to get activated first, and then you'll find that you'll have a gradual recruitment of larger and larger number of motor units. So we'll look at that a little bit later, but this helps us to determine whether we need to say shake someone's hand lightly, or we need to actually crush their hand, right? And so this gives us the ability to have that kind of fine control for how much force of contraction we're going to have there. So again, graded contractions at varied contraction strength based on the number of motor units being stimulated. So that would be just another name for that. So again, be graded, so we can kind of go from very, very soft contraction to very strong contractions. So also looking at this, we'll see that somatic motor axon again coming from the anterior horn, and this is going to be the neuromuscular junctions. We'll look at what causes stimulation there. We'll look at how the actual action potential will end up activating these fibers. Okay, so again, the neuromuscular junction, here's a good picture of one example of this. So again, the neuron coming in, and you notice you kind of have these clefts here within the membrane that allows for kind of invagination of the neuron, and this allows for a very good connection here. They're not really touching per se, but they have a good appropriate approximation to where when they're releasing things like neurotransmitters, they can activate the muscle very easily. You see a lot of these kind of different clefts that are here, and what do you think that is the purpose of having all these? Yeah, we want to have a good increase in surface area. So that way, if we had, say, a lot of release of something like acetylcholine, you can get a very strong contraction versus maybe a weaker contraction if you weren't releasing as much. So having a lot of surface area there helps us to provide that kind of fluctuation. But again, we'll find that when you're stimulating skeletal muscle, we're going to find that acetylcholine is going to be the primary mover and shaker here, right? So this is going to be stimulating nicotinic receptors. Okay, we'll talk a little bit about neuromuscular blockade, and we'll talk about paralytics. All right, so if you ever hear the term neuromuscular blockers, those are paralytics essentially, and we'll look at how those are going to function based on its activities with acetylcholine, either mimicking it or in potentially blocking its activities. But acetylcholine is going to be the really important thing here. Again, another name for this would be the synaptic cleft. It'll be releasing acetylcholine onto the membrane here, and that will cause either an action potential to occur or maybe some other activity to happen there. Mostly action potential. Propagation is a big thing here. Okay, so the general mechanism for muscular contractions. We're going to go over the individual steps, and then we'll look at these more in detail and how we can have some control over these. So looking at action potential, is going to be coming along the motor nerve to its ending on the muscle fibers, right? So that's kind of the picture we're just looking at with the motor end plate. So each ending, the nerve is going to be secreting small amounts of acetylcholine. Small amount of acetylcholine gets released. It's going to hit those receptors and cause the actual action potential to happen here. So basically what you see is that there's going to be acetylcholine-gated cation channels. Right, so we talked about ion-gated channels before, and in this case you're going to find that acetylcholine binds to them, and when that binding occurs, you're going to have an opening of these cation channels. So what do you think flows through these cation channels? Yeah, sodium is going to be the big one, right? So we'll look at where calcium is going to be more important influx-wise for contraction of smooth muscle, but specifically for skeletal muscle, we're going to see that sodium is going to be much more important here. Okay. So anyway, so you're going to open up these cation channels. Sodium is going to flow in, and that's going to trigger and right. So we're going to have the action potential kind of transfer from the neuron over into the muscle, and that's going to cause a depolarization, kind of a spreading wave of depolarization. Again, this is happening at multiple parts in the muscle all at the same time because, again, that neuron is being affected all by the same action potential. So as it branches out, it can affect different sections of the muscle. So again, a lot of sodium to flow through into the muscle, and this will cause that kind of spreading wave, as I mentioned. And so then we have an action potential. Okay, so then what happens after that point? We're going to see. So basically, it's going to travel along. And then this is going to be the same process you would see with nerve fibers, right? So there's going to be basically a nice flow of sodium, and the action potential will spread across the muscle. We're going to look at the T tubules and how those are very important as well for helping to make sure kind of the deep recesses and the deep fibers of the muscle gets contracted as well. But when you have that depolarization, you're going to find that the electricity is going to flow all the way through the center of the muscle, and here it's going to cause that sarcoplasmic reticulum to release a lot of calcium. So this is where calcium becomes important. Not necessarily due to the sodium influx, but there's some other voltage-gated receptors get activated that allow for calcium to be released. Okay. So, and then at that point, calcium is going to allow, it's going to act with troponin. We'll talk about troponin, and that's component of the actin filaments in just a few minutes. But essentially, you're going to cause active sites to become available as calcium interacts with troponin, and then you're going to end up having actual muscular contraction to occur there. Okay. Once that's done, though, and if you left calcium out in the sarcoplasm for too long, what do you think would happen? You just have continued contraction of the muscles, right? So it's really important that we get rid of the calcium as well. So that way, when we want to have an action potential, we want to have a muscular contraction, we have it for the period that we need it, and then we get rid of it, right? And so our skeletal muscles good at performing kind of long-term contractions for a long period of time, like sustained contractions? Not typically, right? So for some muscles, you're going to find that they are very good at having some sustained contractions. Otherwise, I wouldn't be able to stand up here for 4 hours today, right? But you're going to find that usually smooth muscles are going to be a lot better at that. We'll look at some of those differences there and why that's the case. But essentially, the skeletal muscle is better for kind of quick actions, kind of short-term kind of contractions, and then kind of goes back down to baseline. So we need that calcium to be recycled, and that gets kind of shuttled back into the sarcoplasmic reticulum, where it gets stored and gets ready for the next action potential to come along. Okay. So at that point it ceases, and we'll look at a disease state where you can have an abnormal calcium channel that will actually allow for way too much calcium to be released in response to certain stimuli. We'll talk about what that is in a little bit later. But essentially, you have way too much calcium, get way too much contraction of the muscle, and so then we'll look at why that can be very, very dangerous for the person, right? Because of all the energy utilization that occurs there. So anyway, so looking at first the impulse transmission to the muscle, we're going to look at neuronal control a little bit later on, once to kind of get the basic idea of how the muscles are going to be working first. But again, looking at the skeletal muscle fibers, they get innervated by these large myelinated motor neurons, and again, myelinated fibers conduct faster or slower? A lot faster, right? So myelinated fibers is very fast acting because again, we want our skeletal muscles to be very responsive to inputs, and they're going to be affected by these motor neurons coming from that anterior horn of the spinal cord, right? So we said what kind of gets transmitted through the dorsal horn? Yeah, all the sensory stuff kind of gets sent through and that'll be really important. We're talking about some of the spinal cord reflexes and how things are going to be coming back through the dorsal horn, sensory information that gets transmitted back through the anterior horn to cause actual change in contraction. But you can find that these branches, when they occur, they can either happen, say, as few as 3 times, 3 branches of the neuron, or it can happen several hundred times, right? So again, if you're thinking about the one that branches, say, like 3 times, you think you'd have very fine control or very poor control of that muscle? Usually pretty fine control of those kind of muscles, right? When you have large branching of those, usually those are going to be for larger muscles where you don't really need as big of fine control. So think like your quadriceps versus like the stapedius muscle in the ear, right? You really don't need a whole lot of control of that, or you can have very fine control of that muscle versus like the quadriceps, which is kind of more of a, in a lot of cases, kind of an on-off thing versus like this stapedius muscle. But anywho, they're going to form this neuromuscular junction at the fiber midpoint. So usually you're going to find that around the middle of the fiber, usually in the middle of the muscle, essentially, you're going to find that's where most of the innervation actually occurs there. And so we're going to call this point the motor end plate. We can also call this the neuromuscular junction, right? So neuromuscular junction kind of makes sense why that term comes there. And so again, these clefts are going to be very useful because it helps to increase surface area, helps to increase the ability for us to have kind of those graded responses based on just how much acetylcholine gets released there. Okay, so acetylcholine comes from basically combining acetyl-CoA plus choline, so that makes pretty good sense there. I had a professor when I was in farm school, he always called it acetylcholine. So if you said acetylcholine, you're saying it wrong. You don't sound very educated, but I still say acetylcholine because acetylcholine sounds silly to me. But anywho, big thing with acetylcholine is it gets metabolized by acetylcholinesterase. Have you mentioned acetylcholinesterase before? Yes, we have, right? Because that's it's a very fast enzyme. It's very good at metabolizing acetylcholine back into that acetyl-CoA and the choline. And so if you have poor activity of acetylcholinesterase, what do you think could happen? Yeah, so you end up having too much acetylcholine around. You could potentially have too much activation of these skeletal muscles, right? So we also mentioned that acetylcholinesterase is not specific for these nicotinic acting acetylcholine receptors, but they're all, or the actual acetylcholine, but it also can affect what other type of acetylcholine sensitive tissue. Yeah, think about the muscarinic receptor. Think about the parasympathetic effects of acetylcholine as well. So one of the big things we run into is from a tox perspective. I mentioned those organophosphates. This is that enzyme that gets inhibited there, right? So think about certain nerve gases. Again, could be a pretty topical thing nowadays in today's world, but the organophosphates were important insecticides, and they actually inhibited this acetylcholinesterase. And so you end up seeing way too much acetylcholine activity. And so that not only affected the muscarinic receptors, and that's kind of the primary thing you think of, where you have just like way too much secretions of every mucous membrane, you have defecation, urination, but the nicotinic effects are really important here as well, because that can cause, eventually what we'll see is it can cause paralysis due to fatigue of the system, which we'll look at why that is in just a few minutes. But again, very, very important enzyme. Some people may be deficient in this enzyme or certain isoenzymes of it, but for the most part, this should be very active. So again, that choline can be recycled by taken up by the neuron, and they can be formed with acetyl-CoA once again and form acetylcholine. So once we have a nerve impulse that travels down the neuron, so it's going through that anterior horn from the anterior horn from the spinal cord, you're going to find that this will stimulate calcium influx into the neuron. Okay, so once the active potential occurs, you're going to have these voltage-gated calcium channels that open up, allow calcium to flow into the neuron, and that's going to start to activate things like protein kinases and other things that will cause these vesicles full of acetylcholine to start to cause exocytosis. It'll cause fusion of the vesicles with the membrane and will cause release of the acetylcholine. Okay. At that point, it's going to be affecting these nicotinic receptors, and then will cause the downstream effects and cause the action potential to kind of transfer from the neuron over into the skeletal muscle. So once acetylcholine binds to those nicotinic receptors, it opens up these acetylcholine gated channels. So you can kind of see here where this is in the closed inactivated state. There's a lot of negative charges here which help to prevent cations from kind of flowing in this direction, get repelled essentially. But when you have acetylcholine binding here, this will open up the channel and allow for better flow of cations. And this one isn't really specific for any particular cation, but if you think about the kind of extracellular space, what's the most prominent cation? It's going to be sodium, right? So think about the plasma, like you have 135 milliequivalent per liter, essentially, concentration of sodium versus everything else is usually in the single or double digits, right? So because sodium is the most prominent one, this is the thing that flows through in the highest concentrations. So once that occurs, then you can have spread of the action potential along the muscle, and then we'll see the other downstream effects of that causing contraction. Now, too much activation here can lead to fatigue of the system. Okay, so you can end up seeing that if you have too much activation of these channels, either the acetylcholine sticks around for too long, or the molecule that activates these channels keeps the channel open, and they get fatigued and they just stay open, right? Which can lead to eventual fatigue of the system, and actually can lead to paralysis that can occur there. So that's one of the things we run into. You guys ever heard the succinylcholine? Anyone in the EMT world or worked in the emergency department? Or like Sux or succinylcholine? It's a very popular neuromuscular blocker or paralytic that we use to cause paralysis and get someone ready for intubation. Basically, it'll kind of paralyze the entire patient, and then allows the muscles in the neck to kind of loosen up and allows to pass in the endotracheal tube. And so what we actually use is succinylcholine, which actually looks like two acetylcholine molecules kind of stick together, and we give this to the patient, and this actually goes and activates these channels. Activates the channels and then just kind of stays there. And by doing that, you fatigue the system and allow it to cause paralysis to happen. What's interesting is if you have initial activation here, what do you think occurs? Yeah, so you can see a contraction. So you can see fasciculations is what we call that. And so if you see, so we call succinylcholine a depolarizing neuromuscular blocker because it actually comes on and activates the channel, opens it up, causes contractions, and then will cause fatigue of the system. And so if you ever give someone succinylcholine, sometimes you'll see these little fasciculations, you have these little muscle contractions that occur due to the initial activation, and they tire out, and then they're paralyzed, right? Hopefully you're breathing for them, otherwise they're not going to last very long, right? Usually you don't want to give this to someone unless you're really ready for breathing for them. Unless you really don't like them, and then that's probably okay. Okay, so anyway, so again, looking at this end plate potential, you can see basically here typical action potential that occurs. So kind of B, line B is going to be the normal action potential that happens. Normally you'd have again, there's no real kind of pacemaker activity here. The skeletal muscles, it's only relying on input from the neuron to cause activation. So again, acetylcholine binding to those nicotinic receptors, causing influx of sodium, will raise the potential, right? Because now we have a more positive inflow to the cell, and that's going to cause once you hit that threshold, that causes the further depolarization. Okay, so you can get the spread. So what's interesting is that you can cause other effects to the nicotinic receptors and lead to inactivation, or basically you'd have a kind of depressed effect here. And so with line A, this would actually be what would happen if you gave curare. Has anyone ever heard of curare before? Anyone heard of like poison dart frogs? This is essentially kind of what they're secreting is this curare, which actually the reason why they will kill animals, or if you have like indigenous peoples using the secretions from these frogs to hunt and things like that, is because it actually blocks acetylcholine. Basically, it blocks the receptors there, those nicotinic receptors, and causes paralysis to occur, right? So basically, you can try to have the neuron sending an action potential, but if the acetylcholine is never able to bind to those receptors, you never have an action potential happen there. So these would be called non-depolarizing neuromuscular blockers, and so we have a suite of drugs that do this for us as well. So anyone ever heard of rocuronium? It's another one you see a lot in the ER for intubation. That basically does the same thing as curare. It actually will bind to it'll bind to those nicotinic receptors and prevent acetylcholine from actually activating. So even though the neuron's trying to send a signal, nothing's ever going to happen there. So the patient also gets paralyzed as well. But you'll notice that you don't see those same fasciculations, right? Because there's no initial activation like you saw with something like succinylcholine. Okay, so that's one way to do it. You can block the nicotinic receptors. The other thing you could do is actually prevent the release of acetylcholine, right? That would also lead to a similar kind of paralysis type of effect. So you guys are familiar with botulinum toxin. Again, you find it in your Botox, either used cosmetically, certainly, but we'll also use it for patients with contractures, right? So they have kind of inappropriate contraction of these muscles, leading to very severe muscle spasms, very, very painful for the patients. And we can actually give them Botox into the muscles, and this will actually cause decreased release of acetylcholine. So if no acetylcholine gets released from the neurons, even though the action potentials are being sent by the spinal cord, you would never see action potential occur here as well. So that would allow for paralysis of the muscle. We call this a flaccid paralysis. Either one of these cases would be a flaccid paralysis, or basically there's no tone to the muscle because, again, they're just not really being activated very well. Yeah. Or diminished tone, I would say, to the muscle. So you can pick up the arm and they would just drop it back down and really have no control of that whatsoever. So multiple ways you can kind of affect that. Again, there's been some cases of Botox going bad. Some people actually using the wrong concentrations. Kind of less scrupulous individuals who are trying to do this for a cosmetic operation and got their dilutions wrong and end up causing way too much paralysis in these patients. And again, the big thing you worry about causing paralysis as far as respiratory failure is what muscle. Yeah, the diaphragm's a big one, right? So if I paralyze that, I'm not going to be able to ever really have a good inspiration, and so that's the big problem that happens there. So again, be careful who you go to for your Botox. Hopefully it's from a reputable supplier. You already went, but I just see those like signs on the side of the road. It's like $10 a unit for Botox, and I'm like, that sounds like not something I want to like. It's like that, and then like $99 divorces. And I imagine it's like the same office you go to, and I don't think I actually want the same people to do that for me, but. Okay, so anyway, so moving on. So there's also other conditions that can affect the neuromuscular junction. So have you guys ever heard of myasthenia gravis? Right, so this is a condition, it's an autoimmune condition essentially, where you have antibodies that are actually attacking these nicotinic receptors, right? So basically you're preventing activation of the receptors because you're having kind of an inappropriate immune response to yourself. Normally, acetylcholine would bind, cause influx of sodium. You have normal muscular contractions versus these antibodies binding here that would end up blocking that effect, right? So no sodium comes in, no muscular contraction occurs. And so one of the kind of the first things you may see is kind of this droopy eyelid. What's the medical term for that? Yeah, ptosis, right? So you end up having kind of a droopy eyelid, and so that could be one of the things. But weakness, paralysis could eventually occur if the condition got severe enough. And so those are usually the things we want to prevent from happening in the first place. And so one of the things you can do, you can actually give someone a test and to see if they have myasthenia gravis. So you have someone comes in like this, other things you might be thinking about are, cerebral vascular accidents. You think about stroke, affecting potentially one side of the face, things like that. Once you kind of rule out the really scary stuff, if you want to diagnose myasthenia gravis, you can give them what we call a tensilon test or an edrophonium test. And essentially what you end up doing is giving a drug that blocks acetylcholinesterase, right? Just briefly, you block acetylcholinesterase, what does that do? Yeah, so it increases the amount of acetylcholine that's there in the cleft, preventing it from being metabolized, and then you can affect more of these receptors, right? So again, not every single nicotinic receptor gets affected every time you have an action potential come along, but by increasing acetylcholine levels, you have further activation, and you should see an improvement of their symptoms, right? If you see that improvement, then you know, hey, this is probably something like myasthenia gravis that leads you down one pathway versus something else, right? If there was some other kind of neuro issue that was going on. And so you can do that. You can treat this either by giving anti-inflammatories. You can give steroids to actually kind of blunt the production of these antibodies and all that inflammation. But other things you can do is give an acetylcholinesterase inhibitor kind of long term, and that will help with a lot of the kind of the symptomatic effects there, right? So just not really trying to have you guys like memorize all of the patho right now, but just trying to get you an idea of like, okay, here's what goes wrong, and kind of illustrates better like when things are working appropriately, what these systems look like, right? Okay. So looking at the actual muscle action potential, so the T tubules, or these transverse tubules, are going to be very, very important for propagation of these action potentials. Because again, normally you'd find the neuron maybe only be able to really kind of affect the surface fibers, but we find that when you have that spread of the depolarization, you can end up affecting all the fibers, even the very deep-seated ones. And so that's occurring because of these transverse tubules. Basically, what you would end up finding is that when you have activation of the action potential, it would spread, and then it would spread not only along the surface here, but it would also be able to go down into these transverse tubules and cause activation of these kind of deep-seated fibers, right? And that's going to be very important for causing kind of holistic contraction of the muscle versus just the surface areas. And so once that occurs, you're going to find that the action potential is going to cause release of calcium. We call this excitation-contraction coupling. So once you have excitation, you have the contraction that occurs there due to the calcium. I'll show you a picture of this in just a second. But basically, we're going to have these voltage-gated dihydropyridine channels. Or these are not channels, these are receptors, I should say, or otherwise known as DHP. We'll talk about dihydropyridine blockers or dihydropyridine drugs a little bit later when we get to pharm, but just know that they have these voltage-gated channels that get activated when you have the action potential spread along, and that's going to be linked to these ryanodine receptors. Those ryanodine receptors are on the sarcoplasmic reticulum, and that's what allows for calcium to be released into the sarcoplasm and causes actual contraction to occur there. Okay. So the picture that's occurring, so say for instance, it's kind of the surface level of the muscle fibers, the motor unit. You can have the action potential spreading along, come down here. Once you have kind of this influx of sodium kind of all throughout this section here, you're going to have this DHP receptor. Again, this is going to be activated by that change in voltage. And basically it's going to cause the ryanodine receptor to open up and allow for calcium to outflow from the sarcoplasmic reticulum. Okay, that causes the muscle contraction, and then when you're done, you need to get all that calcium back into the cell as well. So this is kind of a different picture, kind of the same thing where you have the transverse tubules here. Again, you have the DHP receptors being affected, and then you have the sarcoplasmic reticulum. Now, keep in mind this is an active process. It does require ATP to shuttle calcium back into the sarcoplasmic reticulum. And why do you think that is? It's against the concentration gradient. You have a lot of calcium being stored in the sarcoplasmic reticulum, so it requires energy to pump things against that gradient and cause it to come back in. But it's very, very important, because if you don't have that occurring, you're going to have kind of inappropriate contractions of the muscle that do not relax. And so that disease state I mentioned before, I don't know if I mentioned it previously, but it's called malignant hyperthermia. You guys heard of that one? Sometimes if you ever hear someone having a really bad reaction when they have anesthesia, it's either due to inhaled anesthetics, or it actually can occur from that drug I mentioned before, is succinylcholine. Basically what happens is that certain patients will have a genetic defect in their ryanodine receptor that allows, when they have presence of these different triggers, like inhaled anesthetics like halothane, things like that, you'd end up having way too much calcium being released from the sarcoplasmic reticulum, and then you just have kind of uncontrolled muscular contractions, right? Because the cells really can't get rid of all that calcium, and so you end up having way too many contractions, and then all those contractions are using up a ton of ATP, and then using ATP and trying to produce more ATP causes what? A lot of heat production, right? So this patient gets very, very hot. They end up developing a lot of CO2s. They get they can become very acidotic in a lot of cases. It can be very, very deadly if you don't catch it and kind of treat it early enough. So that's why if you have someone who's under anesthesia, you're monitoring things like their end-tidal CO2. You're measuring the amount of CO2 that they're producing in their expired gases. Otherwise, you can if you start to see that start to spike up, you would know, hey, okay, something's going on here. You're looking at the patient's temperature, either with like a bladder probe or something like that, and you can say, okay, patient's temperature is going up. The CO2 is going up, they're probably having a bad reaction to this. And so that's why history is really important. You make sure you ask beforehand, like, hey, has anyone in your family had a bad reaction to anesthesia before? And if they do, then that's kind of a trigger to say, hey, well, maybe we should try using a different agent that's safer, or something like that. And so basically the way we treat that is either to cool the patient down, so we use a lot of like cold fluids. We give them a lot of sodium bicarbonate, because that's going to do what to the blood? Yeah, it's gonna reverse the acidosis hopefully and bring them back up to a normal pH, and then we'll also give them a drug that actually blocks the effects of those ryanodine channels, right? So you can actually block them from releasing more calcium, and it allows for relaxation of that muscle, right? So that can be one thing we can do to help treat them. Have you ever heard the drug dantrolene? That's one we use for that. Again, you have to remember these specific drug examples. I would put it on the test if I was going to include that. I would say, like, hey, a drug that blocks the nicotinic acetylcholine receptors in the muscle would cause which effects, right? So you may see something like paralysis or muscular contraction followed by paralysis, something like that. And so you need to kind of go through those kind of steps in logic. But I'm not expecting you guys to know the difference between rocuronium and succinylcholine at this point. Next year, certainly you will, right? Because again, we'll learn all that in pharm. So if you guys are excited about pharm, yeah, no? Okay. You guys said it was totally cool on the review. The pharm is the coolest. I remember at least half of you said that, so I'm holding you to it. Can't take that back now. I have it on video. Once it's on the internet, it's there forever. Okay. So anyway, all right. So let's go ahead and take our 10-minute break now, and we'll come back and talk about the actual mechanisms for contraction. Okay, so again, we covered the basics of the fibers here within the muscle. Again, you can have the large the sarcolemma is going to be covering here all these different fibrils within the cell. Again, these are going to be the nucleus you can find here on the surface, and then a lot of mitochondria, and they're going to be located within the sarcoplasm there to allow for energy production, right? So we'll talk about that a little bit later. But essentially in the myofibril, this is kind of the smallest unit here, is where we're going to find the actual muscular contraction occurs, right? So we mentioned that we have like our Z discs, and Z discs hold what? They hold actin, right? So we're going to be holding actin together. And then we're going to have our H zone. We said, what's the H zone? Yeah, so we just have myosin. There's no real coverage of, there's no overlapping here between actin and myosin. Certainly for this whole A band, there's going to be some part of it that's going to be made up of that H zone where there's no overlap, but certainly for part of it, you're going to find some overlap. You can kind of see it here. It's hard to tell on this picture, right, specifically, but you can notice a little bit of overlap. Those light blue lines are the actin versus the dark red lines are going to be the myosin. So there's a little bit of overlap here in this the A band versus the I band having no myosin, so it's going to be much lighter in color. And so the point here, we're going to have the sarcomere, which is going to be the distance between two Z discs, right? And so this is going to, when you have contraction here, is where we're going to see this area is going to shorten. In some cases, the H zone may disappear because you may have enough of the actin crossing over the myosin, it just disappears completely, or just gets shorter. But basically, this whole distance between the Z disc should get shorter as you can cause the actual contraction to occur, right? So again, we already mentioned this, but I bands contain only the thin filaments. A bands are going to be contain the myosin with some coverage here with the actin. H zones are going to be at that center there of the A band that have no thin filament overlap. I mentioned these M lines. Do you guys know what these M lines are? I didn't mention it on the last slide, but basically it's just the center portion of the myosin, where the myosin kind of gets kind of locked in place, because you notice it kind of spans the center here, but the M line is going to be where those kind of all are kind of anchored in. But again, Z discs are going to be found within the center of each I band there, and again, this is going to be the sarcomere is the distance between two different Z discs. Okay, so looking at the sarcomere, we're going to find this is the basic subunit here of the striated muscle contraction. We're going to notice there's multiple sarcomeres kind of running along the length of a myofibril, and again, it's going to be from one Z disc to the next. And so you can kind of see here this M line. This is also where we find creatine kinase. What do you think creatine kinase does? Has a phosphate group 2. Yeah, so creatine, right? So creatine kinase adds phosphate. So we're going to find that phosphocreatine is going to be a very important molecule for energy utilization within the muscle itself. So that's kind of where also is going to be contained here within these M lines. This is also a byproduct of creatine kinase. The breakdown of phosphocreatine is going to be, do you guys guess? Found in the kidneys. Yeah, creatine is going to be a byproduct of that. So again, when you see breakdown of phosphocreatine, one of the byproducts is going to be creatinine, so it gets produced within the muscles. And then also when you have a lot of muscle breakdown, so you end up having a lot of lysis of these myofibrils, you can find creatine kinase or creatine phosphokinase is another name for this. You can find that within the blood. So if you have someone who has rhabdomyolysis, one of the first lab tests you order is a CPK or creatine phosphokinase. You can measure that and determine how much of that they're really spilling out into the blood. Okay. So again, anyway, you can see the Z disc here, how it's going to attach to those actin filaments. Again, they have that tighten there that kind of performs a nice elastic function there, kind of like a spring. So as you contract those Z lines together, those kind of get loaded, and then when you have a relaxation, it will kind of cause it to go back out to its normal length there. Okay, and so when the muscle contracts, the sarcomeres end up shortening. So you can kind of see the difference here between the relaxed picture here versus the contracted picture. Notice here you're going to have you're going to be pulling the actin in from both directions. It's going to cause a shortening of the sarcomere, and that's when you have your actual contraction there. So the muscle in general gets shorter during that process. Again, there may be a little bit of overlap. There may not be, depending on the force of contraction, depending on kind of what several factors, which we're going to see in just a little bit. And so the H zone, again, either shortens or just disappears completely as you have the actin actually kind of overlap one another. We'll talk about where the optimal areas for contraction are, and where you can see how force production actually can diminish if you get too short in the sarcomeres, right? So there's going to be kind of a sweet spot where you can kind of maximal force, but once you kind of overshoot that, is we're going to see some issues there. We'll also see the kind of increased length of the muscle and also diminish the amount of force that you can produce as well, but we'll look at that a little bit later on. So looking at the actin and myosin a little bit more in detail. So the first thing we'll look at are these the myosin. These are going to be the thick filaments here within the myofibril. Notice here you'll have these two heavy chains. They're going to kind of form this double helix that occurs, and you'll have these two light chains that are going to be on either side. So these kind of form the heads of the myosin. This is what's going to be forming the cross bridge with the actin. Okay, so when we actually had muscle contraction, notice there's going to be an association here between the heads of the myosin attaching to the actin filaments. And so you'll notice that the heads are going to have these ATP binding sites. These are going to be really important. So what we'll find is that when we are ready to cause that cross bridge to occur, there's also going to be an enzyme called ATPase, right? So ATPase, as you might imagine, does what? Breaks down ATP into ADP and the phosphate, right? Absolutely. So you guys, pretty straightforward. But when you cause ATPase activity to cause that breakdown, this is going to get those heads of the myosin into kind of a cocked or a ready position, right? So we're going to find there's conformational changes that occur based on whether ATP is binding to the head there, or if it's going to be ADP plus a phosphate group. But we'll look at that process in just a little bit. But that's mainly where we're going to find these bindings that occur there. You need the ATPase enzyme there, otherwise you're not going to have a good muscle function. And so we'll find that the big differences between smooth muscle and skeletal muscle can be somewhat attributed to the activity of things like ATPase and can affect things like the cycling of these cross bridges. It can affect the speed and the rapidity of which these occur. If you had to think about on a kilogram per kilogram basis, what do you think is stronger, skeletal muscle or smooth muscle? Actually, smooth muscle can actually produce more force on a gram per gram basis there. So one thing you may not always think about, but a smooth muscle is very, very strong contracting. But again, we just don't use it for a lot of movement of things like the bones and whatnot. Anyway, yes, ma'am. Mm-hmm. Yeah, so think about these. So again, think about this here. The heads, the myosin filament are the same things as right here, right? So those light chains, the heavy chains are kind of attaching and forming that cross bridge that occurs there with the actin. So you see actin here. You see the myosin heads kind of binding to it, right? So this would be the center. This would be the H zone where there's no, there's kind of no overlap here, and the M line would be kind of in the middle here. But again, they all have these hinges, and by causing changes in the conformation of those heads, they're going to be moving the actin and moving it more medially. Okay. Looking at the actin, you can see here that it's going to have this double-stranded F-actin protein. There's going to be the light-colored strands that are here. Again, it kind of forms this nice helical structure. Here's an actual electron microscope view of some actin molecules. I just thought it was really funny. But this is what happens when I'm doing these lectures late at night and running on very little sleep. And I think, oh, that would be a fun Google search. Twisty churros. Anyway, so anyway, these you'll also include these active sites, and so the active sites are actually going to be ADP molecules. You can kind of see these little spots here along on the end, and these are going to be sitting on those the double-stranded F-actin proteins. We're going to be sitting on those light color strands essentially. You're also going to have another strain here called tropomyosin. These are going to be the dark colored ones in the picture here. And this is going to be in the uncontracted state or the inactive state. You're going to find that the tropomyosin is going to be covering up those active sites. Okay. So again, if you don't want to have skeletal muscle contraction, you need to cover up those active sites. Otherwise, the whole process is just going to start going, right? The whole process is contracting essentially. So normally tropomyosin is going to bind those up, or at least cover them up, and make it so that they cannot interact with those myosin heads. Okay. And then we're going to have troponin, which you can see the troponin complex here sitting on top of these strands. And basically, you're going to have three complexes there. You're going to have troponin I, which is going to have a strong affinity for actin. So you're going to be sitting on those light strands. You're going to have troponin T, which is going to have a strong affinity for tropomyosin. And then you're going to have one that's going to be troponin C, and so this one actually binds to calcium. So you guys remember when you have the action potential spread along the skeletal muscle, you cause release of calcium from the sarcoplasmic reticulum. This is where it's going to be interacting with this troponin. You guys ever heard of troponin before? What's the context for it? Yeah, usually in the heart, because you'll have a different version of it that gets released. And normally, when can you measure that in the blood? Yeah, during a heart attack, when you have actual damage of the tissue and you're releasing troponin, so one of the things you'll measure there when you're trying to assess if someone is having a heart attack or not, in addition to EKG and other blood work, you can check things like troponin. But basically, when you have this binding of calcium to the troponin, you're going to have a conformational change that will basically cause the tropomyosin to shift. It will uncover the active sites, and this will allow for those cross bridges to form between the actin and the myosin. Okay, and this is going to kind of initiate the process for causing contraction. Yes?
So the troponins are creating a conformational change to pull the tropomyosin away from the actin?
Yeah, so basically normally the tropomyosin is kind of like laying on top of the active sites, and then this kind of pulls it away, so it makes them available to interact with the myosin heads. So there's this walk-along theory, this kind of ratcheting theory for how contraction actually works. And again, some of the stuff in general may be a little bit more on the theoretical side, but this is our best kind of understanding of the process here. But essentially, you find that the actin is only activated when the calcium interacts with that troponin and causes the kind of uncovering of those active sites, and that allows for the myosin heads to attach to these active sites. You can kind of see the process here where the myosin heads can be binding to the active site. When this occurs, you're going to have this myosin head tilt, right? So you can kind of see this process here where you have this what we call the power stroke. It's going to cause the conformational shift. It's going to move the actin filament more immediately towards that M line, or trying to shorten up that H band essentially. So you're going to be moving that across, and then you'll find, we'll go into the details of how this happens, but you're going to have basically a dissociation between the actin and the myosin. You're going to have kind of a resetting of the myosin head, and then you're going to have a reattachment again, right? As long as calcium is available, as long as you're causing uncovering of those active sites, you can have kind of this binding, moving the actin along, unbinding, resetting, and then doing the whole process over again, right? So this is why you can see that if you had way too much calcium available, this process would just kind of continue indefinitely and cause inappropriate contractions, very strong contractions that can occur. They can be very painful for the patient. We're going to see that's when you have muscle breakdown, a lot of energy being utilized, and heat being produced, and acids, and all kinds of other bad stuff. So again, that's why we want to prevent things like malignant hyperthermia from occurring there. But again, these cross bridges can operate independently, so you're going to have a lot of different contractions all happening at the same time. So you can have basically kind of again, you have a uniform kind of movement of the actin filaments immediately, but again, it's not all happening necessarily with the same heads working in unison necessarily. They're kind of working independently of one another. But the more you have active at one time, the greater the contraction force is going to be, right? So the more calcium available, binding to troponin, the more likely they are to have the activation of more myosin heads, more movement of the actin filaments. So we'll talk about, I'll show you a picture of what's happening here in a second, but I think it's good just to kind of like stay out in words kind of what's happening here, this whole process. So I'll show you this in the next slide, so you can kind of refer to that when I'm talking about this. But basically the role of ATP. ATP is very important for muscular contraction, but maybe not in quite the way you would think of it, not in the normal process you would imagine for kind of other cellular processes. But basically what's happening in a step-by-step process, before the contraction, you're going to find that the heads of the cross bridges are going to be binding with ATP. So the myosin is going to have ATP bind to it, and then you're going to have ATPase activity cause that cleavage of ATP into ADP in the phosphate. Okay, this is going to change basically the conformation of the myosin head. This is going to be that kind of cocking process where it's going to get ready for when it binds to the actin, it's going to cause the movement to occur. Okay. But again, it's storing up energy here, right? So in this case, if you have the ADP and the phosphate just kind of sitting on the myosin head, it's not really utilizing energy at that point. The energy is already stored. It's kind of like pulling the hammer back on a gun. It's ready to go whenever you are, right? Whenever calcium is available. So anyway, when the troponin and the tropomyosin complex binds with calcium, that causes the active sites to become available, and then you're going to have the myosin heads are now available to bind with those active sites. They're basically able to attach to one another. And then once this occurs, it's that cross bridge we mentioned. And so then what we're going to find is that there's going to be conformational change in the head. It's going to prompt the head to tilt forward. The arm's going to kind of move forward along that hinge. It's going to provide that power stroke for occurring, right? So basically the head and the hinge are going to be moving, causing the power stroke, and then you have the actin kind of moving immediately, shortening up the sarcomere. So again, that energy that's activating that power stroke has already been stored. So again, that's already being stored there with that phosphate and the ADP that's there on the myosin head. Okay. So once the head tilts, once that cross bridge tilts, we have movement of the actin. You're going to find that you have release of ADP and the phosphate ion. Both of those are going to dissociate from the myosin head, and this is going to cause basically another conformational change to occur. And this is going to allow for basically ATP to now bind to the myosin head. Okay, ATP binds to it, and this is going to cause a dissociation. It's going to cause it to kind of become, cause it to dissociate, move back kind of into its original conformation, and then you're going to have ATPase again, cleave that ATP and ADP and phosphate, and then you're kind of ready to go for another contraction cycle, right? That makes sense so far? Makes more sense when you look at the picture, I think. So basically what you're seeing here is the active site on actin is going to be exposed as calcium binds to it. You can see where calcium kind of comes along. It's going to be binding to the troponin there, and then you're going to see the tropomyosin kind of move out of the way. Now you have all these kind of, you see these little indentations, these little blueberry looking things, and these are going to be the active sites where the myosin head is going to be attaching to. Notice here at this point you already have ADP and phosphate being stored on the myosin head. So ATPase has already done its thing. You've already utilized that energy. It's kind of in a stored state, ready to go whenever the active sites are available. The myosin head is going to form a cross bridge with the actin. It causes here, and then during that power stroke, once you have this kind of shifting where you have the kind of the head coming forward, the hinge kind of moving forward, you have the actin moving along, so you can kind of see where the difference is in position of the actin filament. That's where we're going to have ADP, and the phosphate group are going to dissociate there. Okay, it's going to come off of it, and then once ATP binds to the myosin head again, this is going to cause it to kind of fall back, get into its original kind of starting position. And then once ADP is formed again from the activity of ATPase, now it's ready to bind to a new active site and cause another contraction. That makes sense so far? Fantastic. So looking at the actual tension that occurs within the contracted muscle, you're going to find that the maximum tension gets developed when all of the myosin is overlapped with the actin. So you can kind of see here in these different states of contraction kind of where the force is being produced, or kind of where the most tension in the muscle actually ends up being produced here. So for instance, if in section D there's no overlap whatsoever, you would imagine there would not be a whole lot of tension being produced there, right? Because again, the actin and the myosin really aren't able to bind to shorten one another. No force can be produced here. And that makes sense because you're going to find that point D is right down here at the bottom, right? So that's not what we want. As you start to have once you start to bring in the actin and start to cover more and more of the myosin, you're getting more and more force that occurs here. So you can see a big uptick in the actual tension. B and C are going to be pretty similar. B may be like a little bit higher there as you have all the binding or all the overlap that's occurring there. But you can see that as well as once you start to have too much contraction of that sarcomere, what actually happens is that those Z discs will kind of go in and kind of crumple the ends of those the myosin. Once you have that crumpling, it's not really binding super well to the actin, and that can actually end up losing some force. So there's kind of an optimal period where muscles will be able to produce a large amount of force. Once they get too short, that's when they can start to lose a little bit of that force there. It's kind of optimal range of movement there. Does that make sense? So again, you have just the Z discs kind of crumpling in on the myosin, causing less force. You also find that muscle length as well, kind of in the opposite direction. So if you lengthen the muscle too far, that can also lead it to produce force as well. So again, looking at the maximum tension is going to develop when the sarcomeres end up being at their kind of resting length. They're around 2 nanometers or so is kind of where they're at their natural position, neutral position. This is where they can form the most contraction there. If you imagine, if you were to stretch out that sarcomere, what do you think that happens to the overlapping myosin and actin? It overlaps less, right? Because you're kind of pulling the actin away from the myosin, so you're having less in that overlap. So you're going to be able to produce less force essentially, and so that kind of makes sense. So if you look at the tension, or you actually look at the length of the muscle, so say you're looking at kind of normal length here, you can see that when you have basic contraction of the muscle, it can produce a decent amount of force here, right? Versus if you were to lengthen that, there's only so much contraction of the sarcomeres you can have, and so because less of the myosin is going to be covered by the actin, you're going to find that you can't produce nearly as much force here. So you can kind of find that it dips down as you have further tension in the muscle due to lengthening of it, right? As you kind of increase the tension on the muscle to begin with just by stretching it out. Okay, so again, that's why. Finding especially with like certain movements, they become more difficult in the early phases of it, especially if they're under load already, right? So if you're stretching out a muscle, it's already under load, it can be more difficult to initiate that movement because you can't force as much or cause as much force as when it gets to kind of a more optimal length there. Again, people go to school just for this stuff, and so this is about the extent of my knowledge on a lot of this subject. But certainly, if you go to like kinesiology or other professions, they would certainly have way more detail on this than I can produce. But Okay, so that's the basics of kind of muscle contraction. So kind of looking at the energetics of it, so how we are producing energy and getting it ready for utilization for contraction of the muscle. We'll look at work output for the muscles. Basically, you can kind of equate this to equaling the amount of load that's on the muscle plus the distance we actually move. So it's considered work the muscle is actually doing there. And so you'll see that most of the energy causing this work to occur here is utilized for pulling the actin along the myosin, right? So most of the energy, most of the ATP is being utilized in that process to kind of sit there on the myosin, cause those heads to get converted over to ADP and phosphate, and then it can cause a contraction to occur. But other things that we're going to be utilizing energy for in the muscles also going to be for pumping those calcium ions. We mentioned that can be that's an active process to move calcium back into the sarcoplasmic reticulum, and then sodium potassium ATPase pump. And what do you think function it serves here in the muscle? Same thing it does in the heart. Same thing it does on the neurons. Reset your resting membrane potential, right? So it does the same thing here, right? So you're resetting your resting membrane potential. It is basically kicking sodium out of the cell, bringing potassium back into the cell, and kind of resetting your resting membrane potential so that way you're ready for another action potential to come along. So if you don't have that, maybe more difficult or less difficult to cause a depolarization, maybe leads to issues with muscle contraction. So 3 main sources of energy that we're going to use for contraction of muscles, and again, these are going to be used to form ATP because we saw that was necessary for the myosin heads to be working appropriately. One of the big ones is going to be phosphocreatine. And again, this is going to be basically that creatine is going to have a phosphate group, a high energy phosphate bond already added onto it. And essentially what you can find is this will donate its phosphate group over to ADP and form ATP at that point. The enzyme that's utilized for that is that creatine phosphokinase, or CPK. And I mentioned that when you have muscle breakdown, this is oftentimes one of the enzymes you're going to measure. It can get, you can have, light rhabdo, where you may only have like a few hundred. I don't remember the units off the top of my head, but basically a few hundred versus, people who have really severe rhabdomyolysis can be in the several thousand, 100,000 CPK in very severe conditions. So definitely want to monitor that if you ever have a patient who's having kind of excessive muscle breakdown. But this is not good for kind of really prolonged kind of sustained contractions, and so you may only find that under kind of maximal load or under maximal effort, this is only good for about 5 to 8 seconds or so of kind of muscle contraction, right? So you kind of have a baseline store of ATP within the muscle. This is only good for, once you use that up, then you can use phosphocreatine, and this is going to be only good for about 5 to 8 seconds or so, right? So it could be good enough for kind of short bursts of energy, short bursts of activity, but not for the kind of long-term uses. Other things we can do is glycolysis of glycogen that gets stored in the muscles. We know certainly that excess glucose can be stored in the muscles as glycogen, which can be utilized through this glycolysis process, and that will actually help to reconstitute. When you do this process, it helps to reconstitute both ATP and phosphocreatine. So it's good to kind of help rebuild or replenish both of those stores for energy utilization. And the other benefit here is you can utilize this during in the absence of oxygen, right? So you don't necessarily need oxygen around to cause this to happen, which is good during what type of exercise? Anaerobic exercise, right? So you think about things where you're not able to produce enough oxygen or get enough oxygen to the body. This is going to be very good for those processes. You'll find that this is better than trying to rely on, say, like ingested carbohydrates and things like that from our proteins and lipids from the body. This process is about 2.5 times more rapid. So that's why it's good to have good glycogen stores, because you have a lot of energy available through that glycogen to break down quickly to produce both ATP and phosphocreatine. And so what you'll find is that this is better for probably about a minute or so, kind of maximal sustained contraction. So it's kind of good for a little bit longer bouts of activity. And then probably the lion's share of energy production is going to be through this oxidative metabolism. About 95% of the energy that gets produced in the liver, in the muscles in the form of ATP is going to be generated from this oxidative metabolism. So this is why you can have kind of short bursts of anaerobic activity. You deplete these stores, you're going to have to provide some oxygen at some point. Otherwise, the muscle's going to run out of energy to a degree. Okay. Again, system is not super efficient, so the actual, if you guys go back to kind of the catabolism of our nutrients, we saw that even when you're digesting food, you're losing some energy already because you have to use some energy in order to break that stuff down, so some of that's already lost is heat. But this is not very efficient, maybe less than 25%. which is why when you're utilizing your muscles, you generate a lot of heat due to the fact that this is a very kind of inefficient process. And so I mentioned, like, if you have someone who has like malignant hyperthermia or like that neuroleptic malignant syndrome, you have way too much contraction of the muscles, they generate a ton of heat. They get very hyperthermic. They also end up getting very acidotic due to breakdown products of a lot of these producing a lot of CO2 and things like that. A lot of it due to that oxidative metabolism, right? Okay. So looking at some of the characteristics of muscle contraction. There's kind of two main forms of contraction that can occur here. We'll talk about isotonic and also isometric contractions. I did not realize that was a GIF, but that's cool. See a little bit of movement. All right. Basically, you can find that with isotonic contractions, this is where the muscle fibers end up shortening when tension is being produced, right? So this is when you actually have an actual contraction. The actual length of the muscle will change in conjunction with the actual tension that gets produced there. And so obviously the greater the load, the greater the force of contraction needs to be in order to move that load. You'll find there's kind of two phases here. You have the concentric contraction where the muscle fiber shortens when the force is greater than the load, and then you have the eccentric contraction. The muscle may actually lengthen despite that contraction there if the load is too great, right? So you kind of see the eccentric contraction here. We're kind of allowing the muscle to lengthen back out, kind of releasing it a little bit. There's still tension there in the muscle. You're just not, instead of, I guess like you go to the gym, you see some of these guys like just dropping the weights on the floor. They're not really having a good eccentric phase. You want them to kind of like set things down nicely and maybe not scream at the top of their lungs. That's why I like to have a home gym. I can do that stuff in my garage and not worry about although the foundation's kind of taking a beating. But whatever. Anyways, you can kind of see here, if you were to imagine, having a muscle kind of attached to kind of a steady platform and attaching a weight to the end of it, when you have this contraction occur here, this is going to be isotonic contractions, okay? Okay. On the flip side, you can have isometric contractions, and so this is where the muscles actually themselves do not shorten with contraction. And again, these are oftentimes it can be under voluntary control in some cases, but basically, even though the length here is not really changing so much, the tension is increasing. So you're still having contraction there of the sarcomere. You're still increasing tension, but the actual total length of the muscle does not change at all. And so you can find that certain muscles are very good at helping with isometric contractions. Some of them are going to be not so great, and that makes sense based on the different functions of the muscles. So imagine like something like the soleus or the gastrocnemius, like I want those to be pretty good at isometric contractions, right? Because I need them to be staying stable for long periods of time. So I can stand up here for several hours and I don't really ever fall over, even though I may want to, right? So if you look at the depolarization, you can notice here that even though the depolarization is pretty short, you can see very kind of prolonged activity in some of these muscles here, right? So again, good for things like, especially like anti-gravity muscles, which when I say anti-gravity muscles, what do I mean? Yeah, things that I can use to stand up, right? So things like your, muscles in the legs, the extensor muscles that help me to kind of stand up against the force of gravity. One time I was reading a book about anti-gravity. It was impossible to put down. But looking at other types of muscles, like you may not need those kind of prolonged series of contractions, right? So if you think about something like the ocular muscles, like you don't really need to have kind of long, prolonged contractions of those muscles, and so you need kind of quick movements. And so those are going to be noticing that their kind of period of depolarization is going to be a lot shorter than something like, say, the soleus, right? And so again, makes sense. Go back to the actual function of the muscle and kind of makes sense why you'd want it either to be very quick or kind of very, very kind of slow acting in a lot of cases. So that goes into both our slow and our fast twitch fibers. And so kind of the first ones we'll mention here are going to be our slow twitch fibers. We'll also call these type 1 or red muscle fibers. Why do they call them red muscle fibers? They have a lot of myoglobin, right? So again, myoglobin, remember, that helps to bind up oxygen. It's very dark red in color, so that's why you get that red muscle. And so again, you'll see the myoglobin concentration is actually going to be higher in these muscles than in some of our fast twitch muscles, as we'll see. Basically, though, you're going to find these cause slower contraction, slower speed of contraction, and typically going to be smaller in diameter. For the most part, and then you can see that these are good for sustained contraction for long periods without really fatigue, right? So think about the fact that they have a lot of myoglobin, they're holding on to a lot of oxygen, and they have a lot of mitochondria to end up causing a lot of oxidative metabolism, so that way you can kind of have sustained contraction of the muscles for a good long period of time versus just kind of wearing out like we'll see with some of our fast twitch fibers. And so, again, a lot of these are in the postural muscles, so think about like your spinal erector muscles, think about like your extensor muscles in the legs. These are a lot of slow twitch fibers. And then you have your fast twitch fibers. I know there's an intermediate variety. I just want to focus on kind of fast and slow twitch. But basically, this is type 2. This will also be considered your white muscle. They typically have lower concentration of myoglobin, so they typically have a kind of a wider color to them than the slow twitch fibers. These are typically going to have a lot faster contraction speeds. So they have very large fibers they're going to be utilizing here. And so you'll find that they have probably the greatest ATP consumption. So again, very fast, strong contractions, but they burn out very, very quickly because they utilize their ATP very, very quickly as well. So again, they're going to typically have more glycogen stores, which is good, so that way they can utilize that glycolysis pathway to generate, energy for again an extra minute or so. But they may not be so great at the oxidative metabolism there. And again, found a lot of the stronger muscles. When you imagine people who are hypertrophying in response to exercise, usually it's the fast twitch fibers that end up having kind of the best hypertrophy that happens there because of the fact that they get utilized a lot in those type of load-bearing movements and whatnot. And so again, you find that they're going to be generating a lot more tension than type 2A fibers. These are kind of their intermediate fibers, but they develop less tension than you'll find in the type 1 kind of slow twitch fibers. So here's kind of a table kind of showing the differences here. I'm not really going to get super in depth on things like Z-line thickness and all of that, but just be aware of changes in kind of their metabolic demand and where they're going to be getting the major source of a lot of their energy from, right? Whether it's oxidative metabolism or whether it's going to be relying on those energy stores from phosphocreatine and from glycogen, right? Okay, so going back and looking at the motor units kind of affecting, we mentioned that what is a motor unit? Yeah, so it's all the fibers that get innervated by one single motor neuron, essentially, right? So we mentioned that some of these can be very precise, and you have very small number of fibers per motor neuron, or it can be kind of more numerous, you have hundreds of fibers per neuron here. But again, remember we're coming out from the anterior horn of the spinal cord, is where most of our motor neurons. We're going to go more into detail on these different neurons and kind of how they are being activated or potentially inhibited, and some of the influences that we can see there. But again, this is good for graded contractions, where you can have more motor neuron units being recruited as you need more and more force of contraction, right? So if I just need to squeeze something very lightly versus squeeze it very strongly, those are requiring more and more motor neurons to get recruited when you have those kind of stimuli. So some of the things you can see with kind of repeated contractions, we'll mention twitches as basically where you can have a muscle that quickly contracts and then relaxes due to a single electrical shock, or basically one single action potential of sufficient voltage. One of the things we actually do for patients, I mentioned paralytics before, right? Because a lot of the paralytics we use, I mentioned what do they do to the nicotinic receptors? So the non-depolarizing ones. Yeah, they block the effects of acetylcholine. So one of the things we want to do for a lot of those patients, we want to make sure they're not too paralyzed. We want to make sure that they are paralyzed just enough for things like, being synchronous with the ventilator, so the machine can breathe for them. They're not trying to fight it, by having, any of their muscles trying to breathe on their own. But one of the things we actually do is we have a little device called a train of 4. And essentially what it is you can put it along with the brachial nerves and basically activate it. It sends 4 action potentials or 4 electrical signals into the nerve. And so basically what you end up seeing is you'll find these little twitches of the thumb, right? So of me being not paralyzed at all, you'd end up seeing 4 twitches. So for each action potential, you'd have 1 twitch to go along with that. Versus someone who's paralyzed, they can have different gradations there. So you can see that they can either have 1 twitch, which would mean they're pretty well paralyzed. They can have no twitches, which means they're completely paralyzed. And so you can have kind of everything in between there. So that's actually one of the devices we use by examining kind of this twitch response to someone who is currently paralyzed, right? Because again, you want to cause too much paralysis because that can lead to some longer issues downstream, but we'll talk about those in farm. And, the twitch though is going to be just a single response to an action potential. But again, that's not super useful for our purposes. Most muscles, especially for standing and things like that, we want to have kind of nice smooth contractions that can occur for a long time. You don't want just single twitches necessarily. So what we see is called summation, and basically we're adding together these individual contractions, and they tend to increase the intensity of contraction. So as you are kind of building upon the previous twitches, you kind of have the summation effect that occurs here. So there's kind of 2 major major varieties of summation that happens. We can either have multiple fiber summation, Basically means we're increasing the number of motor units that are contracting simultaneously. So we mentioned that usually smaller fibers or smaller motor units tend to be activated first, is where you kind of get your light contractions, and then as you start to increase the number of motor units that you are stimulating, you get a bigger force of contraction. So that's one form of summation that occurs. There's multiple fiber summation, and then you'll find that there's also frequency summation, where you have an increased frequency of the actual contractions. You have an increased number of action potentials stimulated in the muscle, and this can lead to eventual tetanization. And what do you think that is? Yeah, so it's like tetany, where basically you just have sustained activity of that muscle. So we'll get a picture of that in just a second. But basically, what you end up finding is that if you're looking at just a single action potential, and then you're looking at the force of contraction within the muscle, one action potential, you see that, okay, you have contraction in the muscle, and then kind of goes back down to baseline. Versus if I were to have some degree of wave summation, where I'm kind of increasing that frequency of activation, so I send two action potentials, and just get a little bit of squeeze there in the muscle, and then get even more force. As I increase that, the number of action potentials I'm sending, you see they kind of keep building on one another. And then eventually I can have this kind of, this is called unfused tetanus, where you're going to find some decrease in the amount of contraction between different action potentials. And then finally we get this tetanization, or this fused tetanus, where you can have a continuous stream of action potentials. You're going to see an increased force of contraction, and then you eventually hit a plateau effect, right? So at that point you're hitting maximal force of contraction within those fibers, and this is being maintained by continuous action potentials. So like when you're standing up and you're forcing your say your quadriceps muscle to keep your leg extended, you're having continuous stream of action potentials causing that to occur. You're not just getting one that says, okay, stand up now, and that's it. You're having continuous output there. And then when you decrease the action potentials, then you have a decrease in the amount of force, and then you have relaxation of the muscle, essentially. Okay. So again, incomplete unfused tetanus, you're going to have an increasing frequency of electrical shocks, and that decreases that relaxation time between twitches. There's still a little bit of relaxation that occurs between, but you're going to basically find that it's not a long enough period for it to kind of go back down to baseline. Versus that fused tetanus, we're going to have just kind of an increased frequency to the point where you just really hit that kind of plateau effect. And again, a lot of this is related back to the amount of calcium staying in the sarcoplasm. So I keep sending action potentials, and I keep having those travel down to the T tubules, and I keep having release of calcium from those ryanodine receptors. If I never give the cell a chance to shuttle that calcium back into the sarcoplasmic reticulum, because it just has continued signals, indefinitely, you're going to find continued sustained contraction, because the calcium is doing what? It's binding to troponin, and that's uncovering the active sites, and that's allowing for those cross bridges to occur. So as long as that calcium's around, you're going to have continued contraction. You're going to have continued activity of the actin and the myosin. Energy utilization is going up because you need the ATP to cause the whole process to occur, right? So basically those are the ways we can have kind of sustained contractions within the muscles, and a lot of it is related back to action potentials and their effect on calcium. Okay, and so you can kind of see that stepwise effect. So you can call this TREP. I'm hoping I'm pronouncing that correctly, but basically as the voltage increases, the number of muscle fibers end up using also increases. So you're kind of incorporating more motor units, incorporating more fibers there. And when you hit that maximum value there is when all the fibers are being stimulated simultaneously. And that's that plateau effect we mentioned there. Okay, so muscle fatigue, certainly, I'm sure I've experienced that at some point. Basically, this is kind of exercise-induced reduction in the muscle's ability to generate force, essentially. And so a lot of times this is related back to depletion of things like glycogen stores. So once I've run out of glycogen, I'm not able to produce a whole lot of phosphocreatine. I can't really produce a lot of ATP. If I'm not delivering enough oxygen, or if I'm not having enough time for oxidative metabolism to occur, that can lead to fatigue there, right? And so especially patients who are in a fasted state, what do you think their muscle glycogen stores look like? Probably not great, right? Because again, you're trying to utilize that blood sugar for other things like your brain function or heart function and things like that. And so you'll find the glycogen stores typically are not going to be super high. And in fasted patients, they tend to have more muscle fatigue associated with that, right? You can also find other things that can affect this. So things like increased accumulation of extracellular potassium that can affect things like the resting membrane potential, especially if you don't have enough time for those sodium potassium pumps to be working to kind of reset the resting membrane potential. That could be one thing that can lead to fatigue of the muscles. Say if you had reduced sarcoplasmic reticulum calcium, can help as well. Lactic acid accumulation and lower pH certainly can do this in addition. So we mentioned as you're trying to utilize those energy producing pathways, lactic acid ends up being produced as kind of a byproduct of that, and so that decreased pH can affect muscle fatigue, and then just lack of ATP in general all can be affecting this. So again, but primarily the depletion of stored glycogen is the main thing that leads to that fatigue. So muscle decline with aging, we have this process called sarcopenia. You guys heard that term before? What does that mean? You have low sarcos? Yeah, so basically it's kind of that natural age-related process where you kind of have diminished muscle stores. You have increasing fat stores that occur over time. Usually end up seeing you have reduced type 2 fibers, and we said what type of fibers are these? Fast or slow twitch? Those are your fast twitch, right? So this is why we like to do like resistance training. It's why if you ever like go to the YMCA pool, you see like the water aerobics that are going on there, right? So trying to help them exercise some of those fast twitch fibers so they can try to hypertrophy them back and get a little bit more muscle tissue back there. And you can also see reduction in some of the capillary blood supply. Obviously, this will affect energy production pretty significantly. You have less oxygen getting over to the tissues, and so that can be helped with things like endurance training. A lot of it can help with help by strengthening the cardiovascular support of the muscles. There can be a big thing. But you also find that the reason why the muscle atrophy that occurs over time in these elderly patients, a lot of it can be due to things like chronic inflammation. Because again, we'll find that when you have kind of chronic stress or chronic inflammation, is that catabolic or anabolic? Due to catabolic, right? So we don't need to worry about putting on muscle if we're in a stress state and we're trying to deal with other kind of disease processes. So that's where one reason why you might see muscle atrophy. You can have increased oxidative stress that occurs here due to reactive oxygen species, and then also the kind of the big things that they can affect are things like physical activity, poor amino acid intake. If they're taking other, if they don't have enough anabolic hormones like testosterone around or estrogen, things like that, and then just motor unit denervation, so it's maybe due to like injury or something like that. So all of those can lead to muscle atrophy. So they have decreased performance, decreased strength, and kind of that sarcopenia that occurs there. So it's kind of just a natural form thing you see with a lot of your aging patients. Okay, and then adaptation to strength training. So we have hypertrophy. This is usually mentioned. The type 2 muscle fibers tend to be, they become thicker, kind of more, just larger cells there, and they're going to be increasing the amount of actin and myosin. Basically, more sarcomeres end up being formed there that allows for the hypertrophy of the muscle, essentially. The number of fibers themselves don't necessarily increase, but the number of sarcomeres that are available to cause contraction typically do. And certainly, atrophy is going to occur with the diminished use of the muscle. So if you don't use it, you lose it in a lot of cases. And so especially like patients with chronic illness, atrophy the muscles is a big thing. This is why things like physical therapy and occupational therapy are huge for a lot of these chronic illness patients, to make sure they kind of retain some of that muscle mass, and they retain some of the ability to use those. Because otherwise, again, once it atrophies, it's very difficult to kind of get it back in a lot of cases. And of course, we can adjust muscle length. So by stimulating by stretching the muscles, this can also help an increase length. And what you actually find is that sarcomeres end up getting attached kind of distally to the tendon side and allow for stretch of that muscle. Again, this is a reversible process. So again, if you're not stretching your muscles enough, you end up leading to inflexibility and things like that, just due to the shortening that can occur. And again, all these are just homeostatic processes based on the load and the stresses you're putting onto the body. So if you need to stretch your muscles more because you're stretching them out, they'll get longer. If they need to get stronger because you're lifting more things, then that will get stronger, essentially. Okay, some other muscle phenomenon that can occur here. I can tell you kind of a funny thing to show you just how dumb your professors can be sometimes. You guys ever see that John Travolta movie, Phenomenon? Yeah, one time I was like going through like the DVR and I was like, Phenomenon, what is that movie? I was just really sitting there thinking like, Phenomenon, what the heck is that supposed to even mean? And I was like, oh, phenomenon, I got you. Okay. That's not phenomenal. Okay, so one of the things we can see, rigor mortis. When does rigor mortis occur? Death. We don't want to see this in our patients, hopefully, unless you maybe go into like pathology or something. So this is kind of this prolonged state of contraction that occurs several hours after death. The body goes into the state of contraction, and so basically the reason why this occurs is due to this loss of ATP, right? So we mentioned that when you have the actin and the myosin interacting, you have that power stroke. What is the thing that causes the myosin heads to dissociate from the actin? Binding of ATP, right? That causes that conformational change that causes it to kind of relax back and allows for ATPase to work. So once you start to not have any ATP around, you cannot relax those muscles essentially. So you see that contraction. And so, because you can't relax those cross bridges, this is actually going to be an important process when we talk about the smooth muscles for those kind of really long sustained contractions. But here, so kind of a common occurrence in death. And then what you'll find is that will naturally they'll relax over time due to the fact that you will have a lot of autolysis of these cells as they have no energy nutrient sources, and it'll start to kind of break down. And then say 15-25 hours later, so just goes to show you how long this can take for the decomposition process to occur. They'll eventually get more relaxed in those states. Another thing, muscular dystrophy. This is an interesting one. We see this in some of our kids over at Nemours. We have a lot of studies that are going on trying to find good therapies for these kids. But essentially, one of the most common ones called Duchenne's muscular dystrophy, or Duchenne muscular dystrophy. And so this is actually only affects males. And the reason for that is because it's actually X-linked recessive. So that means that if you're getting it, you probably got it from your mom. So moms can be carriers for it, but you will typically find that for female patients, like they're going to have basically a dominant one that's not going to have those mutations there. But essentially, male patients are going to be affected. And so you have mutations in dystrophin protein. So this is really responsible for doing things like causing that muscle cell stabilization. It helps with changes the way calcium ends up getting handled, cause all kinds of different changes in how the muscle actually functions. And so you end up seeing all kinds of issues here where basically they can end up having a lot of muscle weakness associated with this. They have a lot of foot drop associated with it, difficulty walking. It can be very, very devastating for them. They oftentimes end up living super late into life because, again, things like respiratory muscles end up getting affected. And they may end up going on to a ventilator at some point to treat this, and usually end up dying from kind of other secondary complications. But just so you can see lots of different kind of muscular mutations and things that can happen as a result of aberrations in the normal processes. Okay, I think I'll give you guys another 10-minute break, and then we'll come back and continue with the neural control of skeletal muscles. Okay, so we talked about function of the muscles, how they're actually causing contractions. Any questions on any of that material? Runs with me so far. Fantastic. All right, so now we're going to go on to how we actually control all of that contraction. What are the signals that get sent? How do we send them? And what all is involved with this? So we'll go through this. We'll talk about the spinal control. We'll talk about the kind of upper levels of the brain control of movement, and then we'll get into some of the smooth muscle and then finish this out. So if we finish a little early, that'll probably be okay, right? You guys are okay with that? Yeah, okay. I figured. Had a inkling you guys might be okay with that. But okay, so looking at the motor functions of the spinal cord, so what we can see is that the cord gray matter is mainly going to be this nice integrative area for the cord reflexes. So we mentioned kind of what's coming through the dorsal horn. A lot of sensory inputs. We're going to see that the anterior horn is mainly going to be focused on these kind of motor outputs, right? So it's going to be innervating the muscle itself. And then we're also going to have a lot of these interneurons that are going to be involved here. You're going to find that especially with a lot of the higher functioning kind of CNS effects on the gray matter here, there's going to be a lot of interneurons that's going to be innervating. It's very rare, or at least a small proportion of actual signals from the brain directly innervate these anterior motor neurons. Usually they're going to be going through a series of these interneurons, which will help with some of these kind of reflex pathways, which kind of helps with inhibitory or stimulatory signals. So there's a lot of neurons here that are going to be involved. And again, we're not going to get into detail on every single one of them, but just realize that this is where we're integrating all this information. So that way we can incorporate spinal reflexes, so things that are happening kind of without our conscious control, and kind of integrate that along with things that are under conscious control, right? And so we'll look at all the different kind of features there, but you see things like the cortical spinal tract or the pyramidal tract, like this is the main kind of thing coming from the motor cortex and the up in the cortex coming down and kind of signaling when we want to have conscious control of some of these movements. So we'll look at some of the other pathways that are there. So anywho, so we mentioned that there are going to be the sensory signals coming in through the posterior or the dorsal roots. One branch ends up terminating within the gray matter itself. This helps us to incorporate a lot of those sensory signals that will elicit some of these cord reflexes, right? And then you're going to find the other branch goes to higher levels of the nervous system, and that's where it can go to that somatosensory cortex. You guys remember covering that way back in the beginning of the class? You guys have repressed those memories already. That's okay. We'll talk about repressing memories in the upcoming sections, and probably the PTSD you guys are developing right now anyway. Anywho, but yeah, so the other branch is going to be traveling up, basically up into the CNS, so it allows us to kind of integrate those sensory signals to kind of coordinate with the other functions of the motor cortex. I mentioned the interneurons, which are going to be responsible for kind of integrating all these different signals and allowing for either excitation or inhibition of different outputs onto the motor neurons. And then the anterior motor neurons themselves are going to be the primary things we're going to talk about briefly here, and then get into more detail a little bit later on. So looking at these anterior motor neurons, there's going to be lots of little graphs and charts like this we're going to be going over. So if I don't go over these in detail enough, please stop me so I can cover them more so. So you're going to notice there's going to be kind of two primary motor neurons that are going to be available here. There's going to be the alpha motor neurons, and there's going to be the gamma motor neurons. Have you guys covered any of this stuff previously? Okay, so this might be some new material for you guys. Good. All right, so basically we're going to find the alpha motor neurons. These are going to be useful for innervating the skeletal muscles. These are activating the motor neuron, the motor unit. So when I say neuronal activation at that neuromuscular junction, I'm primarily talking about the alpha motor neurons. This is what's causing contraction of the muscles to occur, right? The alpha motor neurons. You're going to find that the gamma motor neurons are going to be more responsible for innervating these intrafusal fibers. So intrafusal fibers are basically where the muscle spindle is going to be living, and the muscle spindle is going to be very, very important for setting muscle tone, right? So when I say muscle tone, what do I mean? Yeah, basically the resting state of contraction. So I always like it when people say, I want to go hit the gym so I can tone up. And I was like, that sounds uncomfortable. Like, you don't want to increase all your muscles contracting at the same time. That sounds painful. Oh, you'd like to hypertrophy your muscles. Okay, I can understand that. So anywho, so the muscle spindles are really important for that. We'll talk about the reflex pathways that help to control what that tone of the muscle is. So that way when you have, say, increased load on, say, a particular muscle group, how the muscle spindle will respond to that and cause reflex reactions, right? So if you guys think about things like getting kind of those knee-jerk reactions, so if you're stretching the quadriceps muscle when you're doing physical exam on a patient, you see the kind of the knee-jerk. We'll talk about that reaction and those reflex pathways there. And a lot of it goes back to that muscle spindle, which we'll talk about here. But basically, the extrafusal fibers are going to be all of the actual muscle fibers we're going to talk about when actually causing the contraction of the muscle. Again, these are going to be the alpha motor neurons. They're going to be responsible for contracting those extrafusal fibers. The intrafusal fibers are where the muscle spindle lives, and you're going to find that these end up being activated by the gamma motor neurons. Okay, so again, just keep those in mind, the distinction between those two. We'll go over more of those in detail in just a second, but you can see here or basically you can have the anterior horn here where you're going to have the output signals from the spinal cord are going to go into and either activate the actual contraction of the muscle. So again, here, like say for instance the motor end plate, we're going to find these gamma motor neurons affecting the muscle spindle. So again, these would be alpha. These are gamma motor neurons, and all that's going to cause changes in reflexes and things like the muscle spindle. And so this is going to be a sensory fiber that actually feeds back into the dorsal horn. You can also find there's going to be what we call the Golgi tendon organ. We'll talk about more of this in detail as well. But this helps us to detect the amount of tension in the actual tendons, right? So tendon tension is detected here at the Golgi organ, and that also feeds back to the dorsal horn. Now, why do you think this is important to detect the tension there in the tendon? Yeah, it doesn't tear, right? So you want to make sure that you don't have too much force, too much contraction there, because again, if you rip out your tendons, you're going to be in for a bad time, right? So we don't want to do that. So this is going to be the kind of the sensory pathway that kind of helps with that as well. So again, all of these sensory nerves are going to feed back through the dorsal horn. Some of that will be just communicated directly back into the anterior horn here, right? Going through some of these interneurons. You'll notice some of these are going to be positive effectors, some of these are going to be negative effectors, right? So you can notice here how, say for instance, this Golgi tendon nerve is going to be coming through, and it will affect this interneuron here, right? So this is going to be an inhibitory neuron where it may release things like, GABA. We'll talk about neurotransmitters in a later section, but things like GABA are a major inhibitory neurotransmitter and can actually inhibit things like muscle contraction. But anywho, you also find some of this will be incorporated into the higher levels of the CNS. Some of it will be related back to the muscles directly through the spinal cord reflex, right? And so this will have either direct or negative effects on or inhibiting or exciting the actual muscle to contract. So again, we'll go much more in detail on this in a minute, but just realize that these are kind of the basic building blocks we're going to be referring back to. So the interneurons in more detail, these are going to be found in all areas of the cord gray matter. These are going to be very small, highly excitable. They're getting multiple inputs from different types of cells, either coming in from the higher levels of the brain, coming directly from, say, like the dorsal horn. Lots of different places. It's integrating these signals, and there's lots of different interconnections kind of happening here, and they integrate all these spinal cord functions. And so, again, I mentioned you're going to find very few sensory signals or signals from the brain terminate on those actual anterior motor neurons. The grand majority of them are going to be directly innervating, or are going to be going through these interneurons that connect to those anterior motor neurons, right? So there's always kind of a middleman here for these interneurons. There's some other rare cases for things like, cortex activation of like fine motor control, like the fingers and things like that. We'll mention that later when we get to that section. So anyway, a couple other cells you'll find, so things like the Renshaw cells, these typically are inhibitory neurons, and they cause what we call lateral inhibition. I think I mentioned this back when we were talking about the sensory effects, our sensory, the senses that we have. But what do you think lateral inhibition is?
It's like when you shut off the other side.
Yeah, so you shut off kind of surrounding signals. So again, when you have activation of some of these neurons, you can end up activating too many of them. And so some of these Renshaw cells are inhibitory in nature, and they help to kind of sharpen or focus the motor signal. So that way we only are moving the intended muscles and not kind of activating kind of surrounding neurons, essentially. So this is very helpful for helping to sharpen, to allow us to be much more narrow focused when we're causing a motor neuron to be activated there. So again, and then we also have these propriospinal fibers. These are typically running from one segment of the core to another, maybe just be terminating in one other segment. Could be multiple segments it's going to be running to. And these are going to help to coordinate movement, say, in various areas of the spinal cord and various limbs. So these are also very important for helping to send different signals to either excite or inhibit the muscles. Okay, so again, we'll see that the proper muscle control requires appropriate sensory information. If you don't have a good sense of what's going on in the muscles, it's hard to have kind of reflex pathways to account for that. And so again, the muscle spindles are very important for this, and then I'd probably say equal degree, the Golgi tendon organs are going to be affected here as well. And so again, these are used for intrinsic muscle control, and then these are going to be for things that we're operating subconsciously, right? So for movements you kind of perform, we don't really consciously thinking about them. These are where a lot of these reflex pathways are going to be housed here. So intrinsic muscle control. You're going to find that also the information coming from here goes to the cerebellum. So do you guys know what the cerebellum is used for? Yeah, so it has to do with balance. Anything else? Yeah, orderly sequencing of movements, things like that. A lot of like learned movements. So we'll talk about that a little bit later on in this section. But the cortex, spinal cord, all that information will get sent up through those kind of ascending pathways and kind of inform those areas of the body, so that way we can make appropriate decisions on what we need to do. So looking specifically at the muscle spindle, so again, I mentioned that the extrafusal fibers, these are going to be innervated by the alpha motor neurons, and then the intrafusal fibers end up getting activated by the gamma motor neurons. Notice here in the center, there's no actin, there's no myosin here, so this actual section is not able to really be contracted, so to speak. But what you will find, so like kind of the peripheral ends here can be contracted by these gamma motor neurons, but the central portion here is really just going to be more sensory in nature. Really what it's going to be doing is detecting stretch. Okay, so as you cause stretch of this tissue here, that's going to increase the amount of sensory output. Right, so it's going to be, saying, hey, it's sensing that the sarcomere is getting longer, versus if it has relaxation, it'll notice, it'll send signals that says, hey, the sarcomere is getting shorter, right? And so this will be integral for a lot of our reflex pathways. So again, so the sensory fibers are going to feed back to the spinal cord, and then once the spindle receptor gets excited by two main things, one of which is going to be the lengthening of the whole muscle, is going to stretch that spindle and excite the receptor. So again, if you were to have, say, an increased load on the arm and cause it to extend, that will cause an increased stretch in the sarcomeres. That's going to increase stretch in those muscle spindles, which will cause activation of those sensory neurons. And then also contraction at the ends of the muscle spindle by the gamma neurons. These are also going to cause the stretch to occur as well. And so it's important that we have both contraction here with the alpha motor neurons and also these gamma. We'll look at why it's important to have kind of balance between these two to make sure that we can have kind of good reflex pathways no matter what the length of the actual muscle is. Okay, so it helps us to maintain that tone kind of right where we want it. Does that make sense so far? So again, no actin or myosin here, just going to be detecting stretch. Increased stretch is going to increase the amount of signal output to say, hey, we should probably do something about this, usually contract the muscle generally. Okay, so you're going to find that there's, as far as the sensory endings go on those muscle spindles, there's going to be primary and secondary fibers that occur here. So you can kind of see an example of what a primary fiber looks like here. Mainly going to be focused kind of on the center of the muscle spindle, and then you can have the secondary fibers, which tend to kind of branch out a little bit longer. They're going to typically kind of surround a larger area of the muscle spindle, and so it's important we have a balance between the primary and the secondary sensory neurons here, because they have kind of two different functions. Basically, if you were to have, say, slow stretching of the muscle spindle, basically what you would see is that there's a proportional stretch that occurs both with basically the increased signal here in the primary and also the secondary. So if you have a slow stretch, both of them get activated kind of in the same proportion. Okay, this is what we call a static response. And again, so both we're incorporating both the signals from the primary and the secondary. And if it's a slow lengthening, we're going to see kind of proportionate increase in both types of fibers. Okay. On the other hand, though, if we have, say, sudden increases in length or sudden changes in length, we're going to find that we have more of a dynamic response, and that's going to be primarily mediated through those primary nerve endings. So if we have, say, a sudden change in length of the muscle spindle, this is the primary muscle fiber or the sensory fiber that gets activated. As the stretch slows down and gets back down to more kind of those dynamic levels, you'll find that the activity of both sensory endings are going to be about the same at that point again. Okay, so this is good for detecting sudden changes, and if both of these are activated in the same proportion, you're going to find that they tend to help with those kind of static responses. Okay, so I think it's important to be able to tell the difference between the two. Yeah, so you need to be able to detect, like, when am I having kind of fast changes, so I can have those kind of fast reflex responses about. And so being able to tell the difference between the two is very important to make sure we are able to have appropriate reflexes, make sure we don't overshoot a lot of those reflexes, right? So for instance, if I were to, say, bat my hand away, like I want to have it come back to the right spot, I don't want it to kind of go back through. And you can see that when people have problems with, especially like the cerebellum, or issues with these kind of sensory fibers, they tend to overshoot and oscillate when they're trying to move the muscles, basically because they end up overshooting one direction, they try to counter that, and they overshoot in the other direction, and so you see these oscillations that happen there. So we'll talk about those a little bit later, but just know that understand the difference between the dynamic and the static responses, and how these different sensory fibers are going to be responding to that. One thing I'm not going to get super into detail on, but there's kind of two different intrafusal muscle fibers here. There's what we call a nuclear bag, which I thought was kind of a cool name, and there's also this nuclear chain, and all of it goes back to where kind of the nuclei are going to be kind of centered here. These are a lot better. The nuclear bags are better for those dynamic responses versus the static responses are better seen in the nuclear chain fibers. Right. So just kind of an aside. I probably won't test on that specifically, but just know the sensory effects based on the type of fibers there. Okay, so when we see a stretch reflex, and this is kind of, I'll talk about this oscillation in a second, but this is kind of what I was referring back to. But basically, if you were to have a sudden stretch or relaxation, you're going to be changing the length of that muscle spindle, and it's going to cause either a reflex contraction or relaxation within the same muscle, right? So for instance, if I have a sudden lengthening of the muscle, what's my reflex going to be? To contract it, right? So if I put a sudden load on the muscle, stretches it out, my body's going to respond by increasing the force against that in order to maintain stability, essentially. So what you see is that the whole point of the reflex functions are to oppose sudden changes in muscle length. We want to maintain that tone right where it's at, wherever its resting tone happens to be, and we're trying to oppose those changes. And so what you see is that initial phase is the dynamic stretch reflex, right? So it's going to be from those primary sensory fibers. And then as that dies out, it's going to leave basically the weaker, kind of more static reflex, both coming from primary and primary and secondary sensory fibers. And that will help to maintain the muscle contraction. So for instance, when you have a sudden increase in length, you're going to have kind of a strong dynamic response to try to oppose that, and then you want to maintain that response with those secondary kind of more static reflexes. Okay, that helps to kind of maintain the muscle contraction right where it should be. So you can see this here, if you're at a sudden stretching of the muscle spindle will be activated, the sensory fibers are going to feed back, and this is kind of the most simple kind of reflex that you can happen here, or have here, where there's just feedback from the sensory neuron basically onto the motor neuron. There's no kind of interneuron connections. This isn't the most common one, but it's kind of the most basic that you can see there. And so when I mentioned with those oscillations, you can see that a patient who had, or say these are like animal studies that are done here, basically you have an animal who has kind of intact muscle spindles versus one that does not. You can see that if you were to increase the load onto a muscle, you can see that we get a pretty smooth kind of response here, right? So you see the stimuli are going to be the same between the two, say 8 per second. This is resisting the stretch pretty well, but there's a pretty consistent kind of graded response to it. It's not very kind of jerky, right? So we see kind of this initial dynamic response, and then you see the static response kind of holds the muscle right where it should be. Versus, like, say I had an animal that was missing those muscle spindles, you're going to find that the body tends to overshoot in either direction. So the length of the muscle, the force of contraction is going to change wildly and not lead to the muscle being very stable, essentially. Okay, so this is what we call a damping process, and helps to smooth out those reflexes so that way it is very smooth, not very herky-jerky kind of in nature. Okay, so the damping process. Any questions so far? Everyone with me? All right. Okay, so looking at the muscle spindles and how they respond in oh, yes, ma'am.
Did you say damping?
Damping, D-A-M-P-I-N-G, not dabbing, not vaping, anything like that, just damping. If I were to take a moist washcloth, I would also damp you, but this is a different type of damping. Not that I would do that to you, but I'm very tired right now. Anywho, so okay, looking at the muscle spindles, how they work with the voluntary activity. So, for instance, if I were to cause, say, for instance, I cause a contraction of a muscle, I cause shortening of the muscle through activation of those alpha motor neurons. What do you think that would do to the muscle spindles, the length of them? They would shorten, right? So they would shorten up, and then what reflex do you think that would cause? It would make me want to relax the muscle, right? It would make me want to extend, try to lengthen that muscle back out by releasing that force of contraction, right? And so we don't want that to happen necessarily, because if we want voluntary control to shorten that muscle, we want the muscle spindle to maintain function. And so this is where the gamma motor neurons are very, very important here. So for instance, if I were to, say, activate these alpha motor neurons, I was going to shorten up this muscle spindle, but at the same time I have gamma motor neuron activation that stretches the muscle spindle on either side. What happens to the total kind of length here within the sensory area? Stays the same, essentially, right? So by contracting the extrafusal fibers and also contracting these intrafusal fibers on the ends here, we're basically maintaining the muscle spindle length at roughly the same length, and so I don't really have any of those reflex pathways occurring there. And then so by maintaining this, it helps to make sure that I can have voluntary control and not have kind of competing reflex mechanisms trying to prevent me from doing that. So again, anything that stimulates the alpha motor neurons is typically going to stimulate the gamma motor neurons simultaneously. So both are going to be happening together. And so this helps to contract both intrafusal and extrafusal fibers. And so by doing this, it's called co-activation. They get activated in the same degree, essentially. And this helps to prevent that receptor portion of the muscle spindle from activating. And so this maintains that damping function as well, where it helps to prevent kind of those oscillations that occur secondary to those reflex pathways. Okay. And so looking at the central control of the gamma motor neurons, this also helps to stabilize joints, okay? So for instance, if you were to say I wanted to stand, right, and so I need to have a balanced effect, say, between, say, the quadriceps and what would be kind of the antagonistic muscle there? Yeah, the hamstring, right? So I'd want to have contraction of both of those muscles in order to maintain extension of the leg, and that way I can stay standing, right? And so basically what you can see is that by increasing activity of the gamma motor neurons on both sides of that joint, I'm going to increase the length of the muscle spindle. And then what does that make me want to do? Makes me want to contract the muscle, right? So if I'm having competing effects from that on both sides of the joint, I have both agonists and antagonists are both contracting similarly. And so what does that do to the joint? Stays the same. It doesn't move anywhere, right? So that's good because I want a nice stable joint around the knee. I don't want to fall over, okay? And so because of that, if I have things like, if I start to, say, bend backwards or if I bend forwards, I'm going to have responses in those muscle spindle fibers that will help to either strengthen the one muscle versus the other to help maintain that integrity there, okay? So you still have the reflexes that are there. They're very useful for those anti-gravity muscles, those kind of postural muscles that help us to fight the force of gravity, essentially, okay? Make sense? That's the sound that happens whenever I make a good point. Okay, so looking at that clinical application of this, so how does this actually come about in the clinical world? This is a very useful tool, especially like for neurologists and things like that when you're looking for, like, say, lesions in different parts of the brain or in the nervous system. Basically, you can assess for that sensitivity of the stretch reflexes. And so what you can find is that based on facilitatory impulses from the CNS, they can either be enhanced or inhibited. You may find that patients will have kind of an increased reflex or kind of a dampened reflex, essentially. And so what that means is, you would find, if you have an increased impulses from the CNS, it causes muscle jerk to be very exaggerated, right? Much more out of proportion than what you were actually doing. So again, this would be a case where if you were striking your rubber mallet onto the patellar tendon, that stretches out the quadricep. So I stretch the quadricep muscle, and then what does that cause to occur? So the muscle spindles will also stretch, and then that sends back a reflex to do what? Contract the quadricep. So again, I can see the knee start to jerk like that. So again, you notice here how you don't have a whole lot of oscillation there. Basically, you get the one kind of strong contraction, and then it's going to kind of balance itself out, right? So it's that damping process there where you don't see the leg kind of go all over the place. It just has the one big contraction. That's kind of it. If you were to see kind of diminished impulses from the CNS, this could also cause the reflexes to be very weak. So even though you're causing that stretch reflex, you may not see a whole lot of response out of that due to, say, different lesions or different disease states within the CNS. And then there's also what we have called clonus. You guys heard of clonus before? What's clonus?
Rapid movement of the foot is usually caused by
Yeah, so basically it's going to be kind of this kind of repeated contraction of the muscle. Usually you look at things like ankle clonus is probably one of the more common ones you're going to be seeing this. But essentially if you think about standing on your tippy toes and then you drop down all of a sudden, what does that do to, say, the gastrocnemius? Yeah, so if I'm on my tippy toes, that's contracting the muscle because I'm raising my off my heels, and as I go down, that stretches out the calf muscles. That's going to cause what kind of reflex? Stretch the muscles, cause it to contract. Yeah, so that's why you kind of see that kind of bouncing motion. We kind of see this as well. So if you had a patient, say, laying in a bed, basically by taking the ankle and forcing it forward, you cause basically a similar reflex, and so you should feel the patient kind of kicking back at you a little bit. So based on, you can see that be exaggerated or inhibited depending on kind of what's going on with the CNS. We look for this a lot with certain overdoses. So if you were to have someone who had overdosed on certain antidepressants, you can actually see increased clonus due to some of the serotonin effects. And so we'll go look at a patient, and even if they're unconscious, you can go and stretch those muscles there by decreasing the angle there. And so by doing that, you're going to feel that kind of kickback. And so if it's kind of really exaggerated, then that would lead you to think these drug effects are actually around versus not. So we do use this clinically with some regularity there. Okay. So then looking at that Golgi tendon reflex, so again, you're going to find that these sensory fibers are going to be innervating right here within the tendon itself. And so these don't necessarily detect changes in the length of the tendon necessarily, but it's more the tension, right? Because tendon length really shouldn't be changing all that much as opposed to the muscle, but you're going to find that it detects that change in tension. And so you're going to find that this also is going to have dynamic and static responses. So this helps with that kind of damping process as well. And so the opposite effect here, though, is that if you have lengthening of the muscle spindle, so lengthening of the muscle, you cause kind of the opposite effect, you cause contraction. Here what you're going to find is you have increased tension on the tendons. You're going to find that's going to say, hey, we got too much tension here. It's going to feed back. And again, here you can notice how this will feed back through the dorsal horn as well. You're going to have kind of positive effects here, right, because this is now being stimulated. This is actually going to activate an inhibitory interneuron. Right, so now I have increased activation of an inhibitory neuron, which is ultimately going to decrease those alpha motor neurons within the anterior horn. And so by decreasing activity here, by having an inhibitory effect, you end up causing relaxation of the muscle. Okay, so that helps to, if you have, say, too big of an increase on load on the tendons, that can help you to relax the muscle to avoid having basically dissociating that connection there, which again is not a good thing to have happen. So again, these kind of interconnections are going to be very, very important. So even though this is having a positive, this is an excitatory kind of response, you're still going to see that it causes basically an inhibiting effect. Okay, so it's important to kind of get those kind of pathways in mind. We'll talk more about this when we get to things like the basal ganglia and some of the effects you can see there. Okay, also you have these flexor responses. These are really important for helping to manage both the agonist and the antagonistic muscles there. So that you have a response that say, hey, I want to withdraw this muscle. I'm going to contract, say, the bicep. What also needs to happen? You need to relax the tricep as well, right? So you need to have that response occur, and they need to occur kind of in a coordinated fashion. Otherwise, you're not really going to have good movement of the muscle there. And so one of the things we'll see is that one of the most powerful ways you can cause this to occur is with actually this flexor reflex or this nociceptive reflex. So again, nociception is what? sensing pain essentially, right? So if you're like nociception, that's basically just being able to detect or sense pain. And so again, having a pain reflex be this nociceptive reflex. And so again, if you think about it, if you were to step on a tack, what would your initial response be? Remove your foot, right? And then what has to happen in the opposite limb? I have to shift all my weight over onto the other leg, and that way I'm not going to fall over, right? So I can reflexively be able to do that. I can consciously tell myself to remove your foot from the tack, stop the bleeding, shift your weight over to the other side. This is all kind of reflex that this occurs. And so basically what happens here is you're going to have this, and that process of affecting the opposite limb is going to be this crossed extensor reflex. So again, what you're having here when you have, say, a painful stimuli to the hand, you're going to have the sensory feedback go back into the interneurons. You're going to cause a excitation of the excited muscle, right? So you're going to cause an excitation here of the muscle you want to use to withdraw. You're going to have a inhibitory action here on the antagonistic muscle, so that allows us to kind of withdraw back. And then you're going to have also this kind of cross reflex, where it's going to go over to the contralateral side. It's going to end up causing basically the opposite effect here. So for in this case, what you would do is instead of contracting your bicep and relaxing the tricep, you're going to see you're going to inhibit the bicep and excite the triceps. That way you're basically going to do like a Heisman kind of thing. And so that would be the way that could occur with, say, like the hands, right? So if you burn yourself on a stove or something, you're going to kind of push away, and that's that cross reflex there. Okay. Of course, that's kind of the very initial phase. Once all those signals get kind of integrated up into the CNS, that's when you're going to start to see other things like, I should probably walk away from this, or let me turn down the heat on this thing, or whatever happens to be in those cases. Okay. So again, those are very important, all coming from that initial kind of sensory input. Again, pain is usually one of the best ways to kind of elicit this kind of response. Okay. So that's the main kind of spinal reflexes. Any questions about that? Okay, now we're going to move up into the CNS, and we're going to see what other kind of control mechanisms that we have here. So first off, we're going to look at cortical and brainstem control, and then we're going to move into kind of more like the cerebellum and the basal ganglia. So we all can see how these have different motor effects. Okay, so the motor cortex and the corticospinal tract. The other name for the corticospinal tract, if you ever hear this, is the pyramidal tract. And then also, if you ever hear of any of the accessory stuff, you're going to call those extrapyramidal. That nomenclature is probably going by the wayside, but if you ever hear like extrapyramidal side effects or things like that, that's usually what it's referring to. Basically here, we're just really referring to the cortex, down to the brainstem, other things like the cerebellum or the basal ganglia, those accessory kind of organs, those are going to be considered extrapyramidal kind of tracts. Okay, so we're going to see how we can integrate all of these together to help us with things like conscious control of movement or even some conscious control there. So anywho, most of our voluntary movements are going to occur within the cerebral cortex as it activates different patterns of function stored in lower areas. One of the things you're going to kind of notice is if I were to stimulate, say, like a single neuron here in the primary motor cortex, does it stimulate one single muscle? Not always, not commonly, in fact. Normally what you're going to find is it will cause stimulation of several muscles, usually kind of coordinated for one type of movement versus the other. We're going to look at some of the differences between, say, the primary versus the supplementary and premotor cortex and how those kind of differ between the two. But primarily what you're going to find is that they will control patterns of movement, multiple muscles kind of being activated kind of in tandem in order to cause whatever type of voluntary movement you've kind of initiated. And so, again, some movement types will synapse. When these signals kind of go down, you're going to find that a lot of them will synapse onto the interneurons, but more common, that's the most common way that occurs. In some cases, you may see some movements that are coming from the cortex will actually synapse right onto the anterior motor neurons. And so if you think about kind of fine control, dexterous control, things like hands and finger movement, that's mainly going to be just from kind of direct, kind of from the cortex directly onto those motor neurons. But that's not certainly the exception and not the rule in most cases. And then when we're looking at the motor cortex, we're going to find it's divided primarily into the primary motor cortex, premotor cortex, and the supplementary motor cortex, right? So primary motor cortex found here. Notice how it's closely approximated with the somatosensory cortex, which we covered previously in section 1. And then we're also going to have kind of the supplementary and then the premotor cortex kind of down here. Okay, so again, all these are going to function in tandem in order to cause most of our voluntary movements. Okay, so again, this terrifying picture here. I've never seen the show Attack on Titan. Maybe, okay, at least one person with me. It kind of reminds me of that. But anywho, don't go look that up because you'll know how nerdy I am. But anyway, basically what you can find is that stimulation of kind of these individual neurons are rarely going to stimulate the single muscle. As I mentioned, usually going to be patterns of separate muscles. And you can notice here this is very similar to what we saw with the somatosensory cortex, and how you're going to find that a lot of the neurons are going to be focused on fine control of things like the hands, the fingers, the facial muscles, things like that. And then, you know, like things like the trunk, the feet, the legs, like those typically don't have as much kind of real estate neuronally speaking, because we don't really need as much fine control there. But essentially this is situated in the same exact way as you would see with the somatosensory cortex if you go with that. And that makes sense because you kind of want those things to be close together so they can send those signals pretty quickly to one another. So again, looking at the premotor cortex, you're going to find that it also has similar topographical kind of organization as the primary motor cortex. And so basically, you know, all the stuff with the hands and the fingers are all going to be kind of closely situated to one another. And the big thing you find difference here from the primary motor cortex versus the premotor is this is going to be better for kind of more complex patterns of movement. So this is going to be good for things like if I need to position my shoulders and hands for a specific task versus just say clenching my fist, right? So this is going to get more complex. This is going to get away from that primary motor cortex. And this is also where we can start to develop those motor images, where basically like in your mind you're saying, okay, I need to walk over to that, walk over to the opposite side of the room, flip the switch, light switch, and then walk back over here. Those kind of motor images and the things I need to do in order to make that movement happen are kind of starting to be formulated here. And so you're also going to find this will send those signals back to the primary motor cortex to start that initiation process by going through that corticospinal tract, or a lot of cases it'll also go down to the basal ganglia. And so we're going to find that's going to be really important. Again, that's part of that extrapyramidal tract. It will help us with kind of more complex movements. So things like, you know, drawing letters, or being able to replicate, you know, things that I'm seeing if I need to trace something, like, you know, those things are going to be kind of managed through that pathway. And then the supplementary motor area is going to be generally causing mostly bilateral movement. So again, normally, say like the left hemisphere of the brain, like the motor cortex, is usually stimulating what? Stuff on the same side or the opposite side? On the opposite side, right? So again, the motor cortex on the left side is going to be stimulating movement on the right side. You'll find that the supplementary motor cortex is more focused on bilateral movements as opposed to just unilateral. And so you're also going to find that this is going to work with that premotor cortex for kind of body-wide attitudinal movements, head rotation, eye movement, things like that. And so you're going to see kind of more complicated features. And kind of what I think about it is the premotor and supplementary motor cortex. I kind of just lump them together essentially. And so again, there's just going to be more kind of complicated movements as opposed to the motor cortex. A lot of that stuff feeds back into the motor cortex, but the more complicated movements kind of originate here. So think about things like the hand skills, like if I need to play the guitar and I know the E chord, like that would be kind of located kind of here, starting at that premotor or supplementary motor cortex, and then move back to the primary. Okay, so this corticospinal or pyramidal tract, basically you're going to find that the extrapyramidal tract is going to be the accessory pathways. So I mentioned basal ganglia, the cerebellum, the nuclei of the brainstem are also going to be things we'll talk about later on. But this corticospinal tract, you'll notice here, is going to be kind of traveling through and eventually is going to cross over when we're in the medulla. And that's going to cause these pyramids to form here. I guess they're sort of pyramid shaped, I guess. I'm not really sure the details of that, but they like to call them pyramids. That's why where we get pyramidal tract to occur. This is where most of the crossing over occurs as it travels down into the spinal tract. Okay, and so we mentioned previously that most of this is going to be traveling in the lateral corticospinal tract. So primarily going to be seeing these in the, that's why in that previous picture we saw the lateral corticospinal tract being the main place where we're getting those kind of either excitatory or inhibitory signals from the brain. And again, these are definitely more discrete, direct pathways. You're going to find that the more direct they are on activating those motor neurons, the better, more detailed, kind of discrete movements they're going to do. So again, think about kind of fine control of the fingers and whatnot. Okay, so again, going from the cortex, we'll go down to this posterior limb of the internal capsule. So this actually goes between the caudate nucleus and the putamen. These are going to be integral for basal ganglia function. And it's important to notice how closely the corticospinal tract and the basal ganglia are kind of associated with one another, which is good because there's going to be a lot of association there, a lot of kind of pathways that are going to be influencing one another through that. Anyway, Once that internal capsule is going to go down to form the pyramids in the medulla, and then we're going to be finding the crossover here, and they're going to find that lateral cortical spinal tract, and there they can primarily terminate on either the interneurons. It's the primary place we're going to connect up with. They may relay with some of the sensory neurons within the dorsal horn, or rarely they can cause direct contraction of the anterior motor neurons, or direct activation and then cause muscle contraction. Okay, but primarily the interneurons is the main thing that's going to get innervated there. Okay, so incoming sensory fibers to the motor cortex, so kind of where it gets a lot of the information from to start incorporating things together to make decisions on how we're going to initiate movements. You can find that a lot of subcortical fibers come from the somatosensory cortex. You'll also find some input coming from things like the auditory and visual cortices, the frontal cortex, again, where a lot of thought processes and things are going to be coming from, all kind of get integrated there in the motor cortex. Going to find some information from the corpus callosum is going to be coming here as well. What does this help us with? Yeah, basically connects the hemispheres of the brain, so we can kind of incorporate information kind of from both sides of the brain into the cortex. You're going to find some information is coming from the thalamus, and so you can see things like cutaneous tactile signals, the joint and kind of proprioceptors and things like that. They kind of tell us basically what are the outside forces in the body right now, what position are the joints and the muscles in, and so that can get fed up through the thalamus. And then the basal ganglia and the cerebellum, again, part of our extrapyramidal tract, gets incorporated through some of these nuclei. And then finally, we have the intralaminar nuclei of the thalamus, and this actually helps to control things like level excitability. We'll talk about that a little bit later on in a different section of this course. So anyway, so some of the different parts of the corticospinal tract, we'll talk about the red nucleus. And so basically, this is kind of an alternative pathway that's available in people, and you're going to find this helps to transmit some of those cortical signals to the spinal cord. Basically, it's found in the mesencephalon, and so it's usually kind of more well developed in other animals and other mammals and such, but it's going to be a little bit smaller in humans. But regardless, it helps to provide kind of some useful features. And so when neurons are going to be traveling via this route, it's going to be called the rubrospinal tract. It's going to be kind of like rubro, red nucleus. And so these can be utilized for more discrete activation of certain muscles. And so you can find that if you were to, like, say, destroy this in a patient, if you destroy the red nuclei, you typically will find they'll lose kind of fine motor control of the fingers and the hands. They can still use their wrist and whatnot, but they won't be able to really kind of do things as dexterously as they would have beforehand. Again, relatively small in humans, but it's important to note this will be used here. And then this will also be important for incorporating some of the signals coming from the cerebellum, as we'll talk about a little bit later. Okay, so looking at excitation of the spinal motor cord control. So again, we mentioned dynamic and static responses. These can also be sent by the pyramidal neurons. And again, you're going to find you actually have, what's interesting is there's a higher proportion of dynamic neurons are going to be signaling from the red nucleus as opposed to more static responses from kind of coming from just the cortex and the rest of the corticospinal tract. Again, that kind of makes sense if you're having movement within the fingers, kind of really, rapid adjustments in length and contraction and things like that. You want to be able to respond to that with this kind of static responses pretty easily. And so looking at the somatosensory feedback to the motor cortex, this helps us kind of control kind of our precision of the muscle contraction, right? So you can see things like inputs from the muscle spindles, our tendon organs, the Golgi organs there, tactile receptors, those all kind of get fed through the somatosensory cortex back into the motor cortex. And then this can help us to enhance or inhibit muscle contraction. So if you imagine, like, if you're holding on to something and you start to feel slipping out of your hands, what's your brain's response? Grab it harder, right? So then that information gets sent through the spinal tract saying, hey, we're feeling this, these proprioceptors and these tactile receptors are feeling that this stuff is slipping. Let me go ahead and feed that back up through the somatosensory cortex into the motor cortex to say, hey, we should probably enhance that muscular contraction there. So that would cause me to bind harder or contract harder and allow me to keep from it slipping out of my hands, essentially. Okay. And so again, a lot of that can also feed back to things like the muscle stretch feedback. So, if you were to basically, not only do you have just direct spinal cord reflexes that can cause contraction in the muscles, but you can also have some signals coming from the CNS as well. And so by doing that, you can get a stronger contraction. You can get more of a strong signal than it would be if you're just relying on the cord reflexes by themselves. Okay. So again, you can kind of see here, I have the corticospinal tract coming down through the spinal cord. You have that rubrospinal tract which we mentioned is going to be more kind of dynamic signals. It's also going to be more kind of discrete movements. And then we'll talk about some of these other tracks as well in just a little bit. So we have things like the vestibulospinal tract. We'll talk about that in a few minutes. Okay, so looking at brainstem control of motor function. That's kind of how mainly the cortex is going to be managing control of the muscular movements. We also have the brainstem affecting motor control as well. So again, the medulla, the pons, the mesencephalon, they control a lot of kind of subconscious kind of things that you don't really need to worry about controlling, right, on a conscious level. Certainly things like control of respiration. Like how many of you guys are thinking about breathing right now? Probably all of you are right now if you were listening, because now all of a sudden your the cortex is kind of involved there. But normally you're just kind of breathing on your own. A lot of it has to do with kind of the midbrain here. And so this can also affect things like our cardiovascular system, can affect GI function, our equilibrium as well. So we'll kind of look at more detail on these different tracks here, these different nuclei, and how these can help to kind of affect both kind of postural effects, kind of anti-gravity muscles. We'll look at how some of these can affect equilibrium as well, and how these are all incorporated to help us kind of maintain good muscle control. So first, looking at the pontine reticular nuclei and the medullary reticular nuclei, you're going to find that these two happen to be kind of antagonistic to one another. So if you're looking at the pontine reticular nuclei and then also the medullary reticular nuclei, these are antagonistic. And basically what you can find is that for the pontine reticular nuclei, those excite anti-gravity muscles. Okay, so that means it's going to cause me to probably stand up, right? So if you need to stand up for yourself, you want to activate these pontine reticular nuclei, unless you're talking about in the figurative sense. And then on the flip side, you're going to find the medullary reticular nuclei causes those to relax, so those kind of end up getting more activated when you want to sit down or lie down or something like that. And so again, these are going to be kind of stimulated in tandem. You want to have activation of both of these, just one kind of being more predominant than the other. So right now, my pontine reticular nuclei is probably going to be a little bit stronger signal right now because I'm actively standing versus you guys. Maybe your medullary reticular nuclei are going to be more active because you're all sitting currently. And kind of see how they're going to be kind of coming down in different portions of the spinal tract. You can see here where the medullary reticular tract is going to be coming in here versus the pontine reticular spinal tract. Notice how these are going to be feeding into those interneurons, which will either stimulate or inhibit contraction of those muscles. Then we have the vestibular nuclei. So these are going to function in association with the pontine reticular nuclei, mainly to control those anti-gravity muscles. And so what does this help us to do? Yeah, stand up straight and also maintain balance. Yeah, so we need to maintain balance. So this is where this comes into play pretty significantly. So again, it maintains our equilibrium in response to signals from the vestibular apparatus. So you guys remember this thing? Yep. So what does, say, the utricle do for us? Yeah, kind of like horizontal acceleration, right? So we're kind of moving forward or backward. You can kind of tell the difference there versus like the saccule is better for up-down kind of acceleration. And then we also have these semicircular canals. And what are these good for? Yeah, kind of head rotation. Remember, those are kind of on 3 different axes, so that way we can kind of detect movement kind of in all ranges. Again, they're all kind of working together within the cochlea, and that's going to help to, and that membranous labyrinth, send those signals via the which nerve? Vestibular cochlear nerve, right? And so that gets incorporated here in that vestibular nuclei. So you can see there's kind of a medial vestibular spinal tract, and there's also kind of a lateral vestibular spinal tract. So both of these are going to be useful for helping me to, if I start to tip over, I'm going to notice, hey, I'm going out of equilibrium. It's going to help me to properly contract and relax the correct muscles, so that way I can maintain my balance, right? Other things will affect this as well. So like, vision can be really important to incorporate as well. So again, when, if you ever think about those VR helmets and stuff, when your eyes don't match up with what your head's feeling, that usually leads to a lot of nausea and vomiting, right? Because again, or if you're, spinning around like on a roller coaster or some sort of carnival ride, like that disequilibrium between your eyes and what your brain's feeling, all can lead to disequilibrium that can occur there. But you guys remember how these function? Remember those hair cells? Remember the kinecilium? Again, you've all kind of repressed that. So again, we'll go over this real briefly. So again, if we were looking, say, at the semicircular canals here, notice that you have the hair cells, and we have this kind of this gelatinous mass of the cupula. And so what's kind of happening here? What causes activation of these receptors? Fluid. Yeah, fluid's moving around, and it does what to the hair cells? Pulls them one direction or the other, and then what does that do? opens up opens up channels, allows things like potassium to flow in, that causes an action potential to occur, right? So again, depending on which way it's going to be flowing, and again, it's all dependent on the kinocilium, the stereocilia, that'll either lead to increased activation of those nerves or decreased activation, depending on the direction of flow. And again, that's why you move your head this way and you have increased activation of those nerves, and then you twist it back this way, you have basically the opposite effect, depending on which semicircular canal you're dealing with. Versus when we have instead of the utricle or the saccule, these are going to be working a little bit differently, because remember, you're going to have this statoconia. Again, this is kind of a higher density than kind of the surrounding fluids, and so as you have movement either in the horizontal or vertical plane, you're going to find that this will also cause movement here as well, just due to inertia, and that will cause activation of these hair cells as well, right? So it will either inhibit or excite those nerves, and again, all feedback and say, hey, this is what's going on with the equilibrium, and here's how we should affect the muscles in order to make sure we can stay standing upright, or whatever function it is we want to do. So everyone, you may go back and review that if you'd like, in order to make sure you have a good understanding of that. Because it may come up on this test, right? Okay, let me give you guys a 10-minute break now. We'll come back and then talk about cerebellar control, and then we'll go into smooth muscle, and then we'll be done. Any questions from the last 3 hours? All right, fantastic. Think about it like you're watching the extended cut of like Lord of the Rings or something. Like, it's just as boring, but somewhat entertaining, so it's okay. It's pretty good. I mean, it's a lot of walking. The Eagles could have circumvented many of the problems. Anyway, anywho, so going into the cerebellum, I did not realize how much I liked the cerebellum until I was reading up on all this stuff, but we call it the silent area of the brain. So if you imagine, if you were to electrically stimulate, say, like part of the motor cortex, what should you see happen? Some sort of movement, right? You should see some kind of muscle being activated, the hand or face or something like that. But on the other hand, though, the cerebellum, if you were to activate it with electricity, you really don't see any actual like movements that occur. So for a long time, people couldn't figure out what the heck it was used for. But now we realize it's used for several features, which are very, very important. And if you lose any of these, it can be very difficult to move around like you want to. One of the things we see is it's very useful for helping with smooth progression from, say, one movement to the next. So if I need to do something like pick something off of the floor and put it back onto a shelf, like that is a series of movements I need to have happen smoothly from one step to the next in order to make it happen effectively. And so the cerebellum is very useful for this. This also helps for making kind of corrective adjustments based on, you'll find that a lot of the sensory inputs coming up from the spinal cord end up kind of synapsing here into the cerebellum. And so based on not only that, but also inputs coming from the cortex into the cerebellum, you can find that it'll say, okay, well, this is the movement that the cortex said to occur. Here's what the sensory feedback is telling me has occurred. Is there a mismatch there? And if so, how can I adjust the muscle contraction to make sure that things sync up a little bit better? Okay, so it's kind of incorporating a lot of these different kind of signals here to make sure that we're moving like we were supposed to, like we had intended to. Again, working for kind of planning sequential movements. And then also, this helps to kind of learn from mistakes. So kind of working along the lines of like muscle memory, right? So the first time you do something, say for instance, if I like handed you a guitar and I say, here, strum an E chord, and you didn't really know what you were doing, you would notice that there's kind of a lot of like feedback to the brain saying, hey, I'm not really doing this right. This doesn't feel right, until eventually you learn how to do it. And so you're kind of making constant corrections to a point where you can do the action very smoothly, very fluidly. You're not really even thinking about it in a lot of cases. It kind of becomes much more subconscious and control. So look at some of the learning circuits that we can use there to see how that's going to occur. But notice kind of situated here, right inferior to the pons and the medulla. So basically the way that you can divvy up the cerebellum, so if you're kind of like able to kind of fold this out here from the back of the brain, you're going to see this made up of several areas, one of which being the vermis. This is where a lot of the control functions for a lot of your kind of axial movements going to be occurring here. So you can see things like the body, the neck, shoulders, the hip are all kind of located here. So this helps with a lot of like kind of posture, making sure you're using those anti-gravity muscles appropriately. So kind of all controlled through the vermis. You can kind of see this here in this picture as well, which you can kind of see the topographical breakdown similar to what we saw like the motor cortex and the somatosensory cortex, and kind of see how things are mainly the actual skeleton and body is going to be kind of maintained here. Versus in the intermediate zone, this is where you're going to find a lot of contraction of kind of distal portions of the upper and lower limbs. So you can see where the hands and the feet are kind of going to be situated here in kind of this in this intermediate zone. Okay, and then you're going to finally have the lateral zone. This one's going to primarily communicate with the cerebral cortex, and this is what's really important for assisting with those sequential movements, right? So kind of going from spot to spot to spot, or going from action to action, is going to be maintained here in the lateral zone as it communicates with the cortex. Okay, so looking at the input pathways, there's going to be several different, and this is important for understanding how the cerebellum is able to kind of take all these different inputs and then output them back out, and for helping with things like coordination, timing, making sure we're judging distance correctly, based on all these different inputs that are coming in here. And so one of the ways you can see here, the first one is going to be this cortico-pontocerebellar tract. Sorry if I say any of those wrong. Basically, this is going to be looking at the cerebral motor cortex, the premotor cortices, and also the somatosensory cortex are all going to be getting kind of feedback into the cerebellum from these different sites. And basically, it's going to be passing by way of this pontine nuclei in the pontocerebellar tract. So you can kind of see that. Where's it at? See the cerebropontine tract kind of coming down here, and this is going to be eventually feeding in here. Okay. You're also going to have this olivocerebellar tract. You guys know where the olive is? Not in your martini. It's in the medulla, right? So you're going to find the olives there within the medulla, and that's also going to be useful for sending some information coming from the cerebral motor cortex. Also, you're going to get information from the basal ganglia, which we'll talk about in just a few minutes, the reticular formation, and the spinal cord. So a lot of information kind of gets all fed through the medulla. We know that the brainstem is responsible for kind of acting as kind of a way station for all of these signals, and so you can see where this can kind of feed into the cerebellum. Also, the vestibulocerebellar fibers. Again, this is going to be very important for helping us with the basically with equilibrium and whatnot, because it's going to communicate with the vestibular apparatus by way of that vestibular nuclei. So we kind of mentioned that previously, where they're kind of situated within the spinal cord. And this also will terminate in that flocculonodular lobe, which you can kind of see down here in the kind of lowest portions of the cerebellum. I didn't mention it on the previous slide, but you can see it there on that picture as well. And then we'll also have these reticulocerebellar fibers. These are originating within that reticular formation in the brainstem and terminates in the vermis. I'm primarily going to focus on the inputs coming from these first three tracks, because those are going to be most important for our functions as far as movement, sequencing movements, timing movements, different things like that. So some of the ways we're going to get information coming from the spinal cord as far as from sensory information, how that will get back up to the cerebellum. We're going to see there's certain afferent pathways from the periphery, and so this is going to be receiving information from several different tracts within the spinal cord, but the two most important ones are going to be this dorsal spinocerebellar tract and this ventral spinocerebellar tract. Obviously my enunciation goes out the window when I've been talking for this long, but stick with me. Anywho, so for the dorsal spinocerebellar tract, you can find that basically runs along this line here, and this is going to be entering the cerebellum through this inferior cerebellar peduncle, and it's going to be terminating in the vermis and also the intermediate zones on the same side of origin. So basically, information coming from the right side of the body will be entering on the right side of the cerebellum. And so we're going to get a lot of information from the muscle spindles, the Golgi tendon organs, and also other skin and joint receptors. So this is where a lot of information about where are the muscles at, what is going on as far as contraction, as far as tension goes, and it feeds back into the cerebellum. So we can start to incorporate, like, okay, well, how is this matching up compared to what the cortex is sending out as far as the voluntary movement? Are things matching up appropriately or not? And so this tells the cerebellum, about the contractions, about the tension of the tendons, forces acting on the body. And so this is where we're getting a lot of that information. On the flip side of that, you're going to have the ventral spinocerebellar tract, and it's going to be entering through this superior cerebellar peduncle, and it's going to be terminating on both sides of the cerebellum. So again, a little bit different there from the dorsal one. And so you're going to see this actually being excited by motor signals in the anterior horns. Okay, so basically you'll have the corticospinal and the rubrospinal tract terminating down on some of these anterior motor neurons, and so based on that feedback, that information that gets sent there, that'll get fed back up in the cerebellum. And so basically, this will help to tell the spinal cord, or tell the cerebellum, what signals have actually been received by the anterior horns. Okay, so that makes sense, because if the cortex is saying, hey, I want to initiate this type of movement, what signal is actually getting down there to the anterior horns? Does it match up with what the cortex has been telling the cerebellum directly? So all that information can feed back up to say, okay, well, are things matching up appropriately? How do we need to sequence things and make sure we're moving like we should be? Okay, so then we're going to look at the output from the cerebellum. So once we kind of incorporate all that information, then we can output it out and see what happens there. Basically, we have these 3 deep cerebellar nuclei. We're going to have the dentate, the interposed, and this vestigial nuclei. Basically, they can kind of help with 3 different functions based on kind of where they're situated within the cerebellum. So this dentate, you're going to find this is helping to coordinate sequential movement. So if you had to think which part of the cerebellum you might find that dentate nucleus primarily. Kind of the intermediate, the lateral, the vermis. Probably that lateral aspect of the cerebellum, right? So a lot of the signals will be coming in there to the dentate nucleus. We're going to call this cerebrocerebellum. Basically, the tracts that they form can be considered that. The interposed nuclei is going to coordinate some of those reciprocal movements of antagonistic muscles in the peripheral limbs. So this is going to be more associated with that lateral area. The intermediate area of the cerebellum, based on if we need to move our arms a certain way, it helps to kind of coordinate antagonistic muscles and contracting and relaxing in appropriate fashions. So we call this the spinocerebellum. This helps a lot with a lot of the spinal cord kind of reflexes we'd be experiencing anyway. And finally, the vestigial nuclei helps with equilibrium, so we call this vestibulocerebellum. So again, obviously it's going to be mainly focusing here closer towards that, the portion I can't even think of the name now. Anyway, it helps with equilibrium. It helps to make sure we can stay upright when we need to. And so again, what you'll find is that every signal that comes into the cerebellum ends up splitting, and one portion of it will end up going into the cerebellar cortex, and the other portion of it is going to be situated into the nuclei. So we'll look at a diagram of that in a second and why that's important. But it's important to realize that these signals are going to be splitting, the sensory signals, and then how they get fed back into an output signal will be very important for us. So this is a fun chart. You kind of make sense of it as we go through it kind of slowly. But essentially what you can find is they're going to have 3 layers for the cortex. You can see the molecular layer here. We have like really short kind of interconnecting interspersed neurons. You're going to look at these Purkinje cell layers. And then we're going to see the granular cell layer, and then finally we're going to have the deep nuclei. So this is kind of the functional organization of the cerebellum. Basically, all of the output signals are coming from the deep nuclei. Okay, so a lot of inputs are going in. Most of the inputs are going to be either outputting onto the deep nuclei cells and also the cortex. Again, the split's always going to occur, but all the output's going to be coming from the deep nuclei cells. So basically what we see happening is that you can have different types of inputs coming in. So you can have what we call basically these climbing fibers, or we can also have these mossy fibers. You can kind of see why they call them mossy fibers based on the way they kind of split and kind of communicate out with the different areas of the cortex here. But essentially what we can see is that you're going to have all the inputs as they come in. They're initially going to be initiating activation or excite the deep nuclei cells. So again, this is just kind of the way the input signals are coming in. They're all going to be activating those deep nuclei cells, right? So you should see initiation of movement that can occur. And I'll kind of tell you how that coordinates with the cortex in just a minute. As the signal continues to go up after the split, it's all going to be feeding back into the cortex here, and that is actually going to a few milliseconds later, you can see activation of these Purkinje cells, and all of these Purkinje cells are going to be inhibitory. So initially what you'll see is you're going to have an activation of the deep nuclei cells via these the split in the incoming sensory information, and then you're going to have an inhibitory effect by the Purkinje cells, just a fraction of a second later essentially. So why do we want to have that? Basically what we can see is that in general, we're going to find that we're favoring output from the deep nuclear cells rather than trying to inhibit. So typically, if you're kind of focusing on the balance between Purkinje cells and the deep nuclei, you're focused a little bit more heavily on the deep nuclei, kind of at a resting state. Okay, and so what we can see is that if you were to have rapid movement of a particular muscle, you're going to find this actually stimulates the deep nuclear cells initially, right? Because again, that's just the first place that the signal is going to be hitting the deep nuclear cell, and then it's going to eventually lead to inhibiting movement. So this kind of helps with that damping process we mentioned before with the dynamic and the static sensory fibers, right? So again, if you were imagining if I were trying to move my arm this way, if I only wanted to say put it right here, if I want to put my finger right here on the desk, I don't want to accidentally overshoot that. And so by doing that, by having those inhibitory processes, I can initiate this movement through the deep nuclei, and then I can inhibit it with those Purkinje fibers. So one of the problems you'll see with patients with cerebellar dysfunction, they can overshoot very frequently. So even though they're trying to put their finger over here, they'll end up overshooting it, and then they overcompensate. And so you can see this oscillation. It can be very difficult for them to perform the motor function they want to do, right? So And then you'll also see that the cerebellum will also strengthen the signal coming from the cortex. So initially you'll find that through the corticospinal tract, possibly the cerebrospinal tract, you'll have initiation of a muscular movement. But you'll also find that the cerebellum is also going to help get feedback from there, get feedback from the corticospinal tract, and will actually send also an excitatory signal from the deep nuclei first. So you actually get a stronger signal to move the muscle initially, and then the Purkinje cells will actually help to stop it. So you have better on-off of the muscles essentially. So you move when you want to, and you stop moving when you want to as well. Okay, I'll talk about some cerebellar dysfunctions in a little bit, and so you can see how that kind of goes awry when that's not working appropriately. The other thing you'll find with these Purkinje cells is they actually learn to correct motor errors. And so what you find that is if you say we're doing something incorrectly, you find kind of increased inputs that inform the cerebellum of this mismatch. Basically, they'll kind of detect, hey, things aren't working correctly. And you may find kind of an increased activation of those Purkinje cells to inhibit that movement, right? As you start to learn the movement and get better at it, you'd actually find less of this kind of inhibitory effect from the Purkinje cells. So that can be very useful for things like muscle memory and kind of learning how to do a process over time. Make sense so far? Everyone just have a glazed look on their eyes. It's okay. That's why we record these things. Gotta make my YouTube monies. Just kidding, I don't make it. I wish I made money off of that, but that would be ethically a little questionable, so I'm not going to do that. So some of the things you can find as far as abnormalities that can happen in the cerebellum. So I mentioned, things like overshoot and that oscillation that can occur, but you can have things like dysmetria and ataxia. Basically, if you were missing that cerebellum, or if you had some kind of lesion there that was preventing appropriate function, what you could see is that one thing you'd be able to not really be able to predict how far movements are going to go. So I mentioned that instead of being able to put my finger right here like I want to, I would end up overshooting and then kind of not being kind of overcompensating. I would have a hard time getting my finger right to where I want it to be. So again, that brain overcompensates when you see that oscillation. So the cortex will say, hey, I missed my mark, based on somatosensory inputs and other things. I'd say, hey, I missed my mark. Let me go back this way, and then I would miss it again, and then miss it again, and eventually I'd be able to hit my mark. Also, that's kind of this past pointing where I mentioned if you don't have that on-off kind of features, you may go past the point where you wanted to stop. And this dysdiadokinesia is actually kind of an interesting thing as well. So this is a thing you can witness in patients where if you're asking them to kind of flip their hands over back and forth, right? So a patient with good cerebellum can be able to say, okay, well, I can just move my hand back and forth, no problem. What you'll find with patients with dysdiadokinesia is they would have an impaired ability to really cycle back and forth. So they may say like, they'd have to, there's a lot more conscious control they need to do that, and so they would not really be able to do that appropriately. They may get it right every once in a while, but they would have really a hard time causing that kind of flipping of the hand, essentially. So that's going to be one of the things you want to look for patients who have cerebellar dysfunction, especially like things like strokes and whatnot, is some of the things you can look for. They can have problems like dysarthria. You guys know what dysarthria is? Yeah, so difficulty kind of like articulating or speaking. So they have a hard time with that sequencing of things like, well, how do I move the laryngeal muscles? How do I breathe appropriately? How do I move my mouth muscles in order to make the sounds that I want? You can tell I'm having a little bit of dysarthria already. But some of that, it gets even worse because they can't really kind of function and sequence their speaking to where they can say the syllables they want to. They may kind of hang up and get caught on one syllable or not be able to kind of sequence things appropriately. So that'd be one thing the cerebellum is really useful for. And then also they have an intention tremor. And so you see there's kind of a difference in the pathophys between intention tremors and then resting tremors. Look at a disease state in a little bit that causes a resting tremor, but basically because they have kind of an impaired ability to do that on-off, because they have problems with only really the cortex kind of managing things, they will start to have tremor once they initiate movement. So be trying to like reach a certain point, they'll end up kind of overshooting. That oscillation occurs there, and a lot of that's all intention tremor. So if they're at rest, no tremor whatsoever, but once they start to initiate movement, that's where you can see the tremor there. So important. We like to keep that functioning as best we can. Next up, we have the basal ganglia, and then talk about motor function, how that is coordinated there. So we kind of see essentially the basal ganglia is kind of located here. Notice how it is very, very closely approximated with the corticospinal tract, right? This pyramidal tract. And so this is important because there are going to be lots of kind of circuits that are going to be feeding from one system to the other to influence our movement. This is going to be made up primarily of the caudate nucleus. You can kind of see the caudate nucleus kind of wrapping around here. Putamen, the globus pallidus, the substantia nigra, and the subthalamic nucleus. So the thalamus will be kind of closely associated with this system as well. And so you can see here, it's pretty complicated. I'm not going to have you guys remember, okay, it goes from this place to this place to this place, because it gets very complicated. And so, but it's very important for appropriate movement, for initiating the movements that we want to, for voluntary movement especially. And so we'll get some disease states where that can be affected there, but just know it's very complicated. There's lots of extensive inputs and outputs pathways that happen here, and so derangement of any one of these spots can lead to issues. Okay. But the two main things we're going to look at, there's going to be a putamen circuit and then also a caudate circuit. So we'll look at those two and how those are kind of useful. So one, basically the putamen circuit ends up getting activated for kind of complex motor activities. And this is, again, an association with the cortical spinal tract. So again, if I needed to do something like write letters on the board, if I needed to cut paper with scissors or hammer a nail, those are kind of complicated complex motor patterns that I need to be able to activate. And so again, just using the corticospinal tract may not be good enough to do those kind of complicated patterns, and so the basal ganglia can be useful for this as well. You notice that doesn't have a ton of inputs coming from the primary motor cortex, and in regards to complicated movements, the primary motor cortex isn't really involved with a lot of complex movements anyway, right? It's going to be more kind of simpler motor patterns. But what we do see is that this process is going to be feeding in from both the premotor and the supplemental motor cortex. The prefrontal cortex is all going to feed into the putamen, and then again through some kind of complex inputs and outputs, you're eventually going to see this will feed back into the premotor cortex, but also into the primary motor cortex. And then once the signal kind of gets fed back into here, now it's able to initiate this kind of complex movements through the corticospinal tract. And so some of the things you can see if you were to have, say, dysfunction in the putamen circuit, say, for instance, the globus pallidus had a lesion in it, you'd have a very difficult time writing. In writing, you couldn't write letters appropriately. You'd look almost like you're trying to learn for the first time, like your letters would look very, very crude. You'd have kind of writhing movements of the arms, the neck, or the face. And if you were looking at a lesion in the putamen specifically, you can actually see this chorea that occurs, kind of these flicking movements that happen. Okay, so again, depending on where strokes occur, depending on other types of lesions, you can see lots of different kind of movement disorders that can occur from this. But again, this is all the putamen circuit, better for causing those kind of complex motor patterns to occur. And then we have the caudate circuit. This is going to be better for more cognitive control of motor function. So think about if I want to have, I want to initiate something, I'm consciously thinking of initiating a movement. This is where the caudate circuit kind of comes into play. And so basically, I should say lobes and not lobes of the cerebrum, but basically the caudate extends through all lobes there of the cerebrum. So you can see that it associates with the prefrontal cortex, and it gets involved with areas of vision and hearing, where a lot of that information can be kind of sent together to help us form that motor picture, essentially, right? So that picture of like, okay, I want to do something, how do I make that happen, right? And that's where this comes into play here. Notice this actually does not output on the primary motor cortex whatsoever. It's going to be more concerned with kind of these sequential patterns of movement. So think about things that are going to take more than 5 seconds to do. So if I needed to walk from here to the light switch, turn it on, come back over here, do whatever on my computer, that would be kind of a more complex pattern. The caudate circuit would be more used here. Another example is if you had, say, a bear attack, say a bear hopped out. The steps that you would need to take in order to get away from that, you'd have to, maybe close your laptop. Maybe you wouldn't be thinking about that. You get to stand up, walk to the center row, run out the door, hit the elevator button, get onto the elevator. So these kind of complex, longer sequential pathways get managed through the caudate circuit. Okay. So who's heard of Parkinson's disease? A lot of people have. This is an issue with the substantia nigra, as we'll see here. But basically, you're depleting the brain of these dopamine secreting neurons that occur here, and so we'll find that dopamine is a very important neurotransmitter for helping to initiate and sustain movement. So what happens is, and I'll show you the pathway here in just a second, that basically causes this rigidity that occurs, right? So has anyone seen the movie Awakenings with Robert De Niro, I think? I really recommend you watch that movie because it's really a good example, or it kind of shows you how the initial treatments for Parkinson's came about with L-DOPA. Very, very interesting kind of movie, seeing how those patients go from basically catatonic, unable to move whatsoever, to having kind of normal lives again for at least for a period of time. But also, these patients will get an involuntary tremor. They have a hard time, they basically will have this kind of pill rolling that can occur. That's kind of commonly heard of here. And then they'll have problems kind of initiating movements. And so they'll have kind of these kind of short, kind of shuffling gaits that they have due to the inability for them to really kind of activate movements on their own. And eventually they can get to a point where even though their brain's saying, hey, make this movement happen, the body's just not going to respond whatsoever, right? You can also get some other speech disorders and gait disturbances and whatnot. The reason why that happens, you can kind of look at here being, this would be the normal kind of pathway. That we're going to go over this in much more detail in pharmacology when we get to the Parkinson's section, but here's basically what happened in a Parkinson's patient. And so you can see the kind of the delicate interplay between both inhibitory and excitatory neurons. So if you're looking here, this being the substantia nigra, we're secreting dopamine neurons, right? So I'm not going to ask you this on the test, but I just wanted you to at least appreciate the complexities here. Basically what happens is you'll find that certain dopamine receptors are going to be more stimulatory. Some of them are going to be more inhibitory, right? So you can find there's different subtypes of receptors, so D1 and D2 in this case. Now you can have a GABAergic neuron. We mentioned GABA is excitatory or inhibitory. It should be inhibitory, right? So we'll go into more detail on that later, but basically it should be an inhibitory neuron. So if I send a positive or an excitatory signal onto a GABA neuron, what do you think happens to the subsequent neuron that it synapses with? Should be inhibited, right? Because I'm releasing GABA onto it. We know GABA is inhibitory, so it should inhibit that next neuron. So this is kind of what happens here, where basically from the striatum, the globus pallidus externa, back to the globus pallidus interna, you're going to have a balance between both GABAergic activity, where you're inhibiting this neuron, and also stimulating this neuron, right? With glutamate. Glutamate is going to be a very important excitatory neurotransmitter. And so by having this balance here, you can see good effects on the thalamus, and that's going to feed back to the motor cortex, and so I can initiate movements when I want to. What happens, though, is when you have patients who have depleted their dopamine stores, usually have a destruction of those neurons, they're not releasing any dopamine here, and so you have less activity of this neuron, because this used to be stimulatory, now it's not stimulated anymore. And then now you have overactivation of these neurons here, we have more GABA coming out, because again, this used to be inhibitory, but now this has kind of unopposed activity, so you're releasing more GABA, this is going to inhibit this neuron, and so initially what you end up seeing is that you have too much glutamate activity onto a GABAergic neuron, which is going to eventually inhibit the thalamus. So you can see, based on even though that I'm sending a signal to say this to move, because there's too much inhibitory effects on the thalamus, it doesn't feed back to the motor cortex, and that movement doesn't occur. Okay, it's complicated, right? But this is why it's good to have an appreciation for the basal ganglia and how it kind of gets incorporated into all of this, to helping initiate these kind of more complex kind of motor patterns. Okay. So any questions on neuronal control of skeletal muscle? Go back, just kind of let it sink in for a while. Try and go back and read it again. The book's actually pretty good. I know you guys don't like reading books, but go back and read the book. I actually thought it was pretty good. All right, moving on. Real briefly, we're going to talk about smooth muscle. Smooth muscle in the past in regards to things like our hollow organs and how those can be affected by smooth muscle. Basically, there's two main types of smooth muscle we'll deal with, and we're going to talk about the differences between this and skeletal muscle and how they contract a little differently. With smooth muscle, you're going to find they can either be in a multi-unit smooth muscle. So again, this would be an example of a multi-unit smooth muscle. Notice all these fibers are kind of discrete. They're separate. They're not really interacting with one another necessarily. You're going to find these are primarily under neuronal control, so usually some kind of stimulus from the CNS will cause changes here. Think about things like the ciliary muscle within the eye, the muscles that control the iris, piloerector muscles within the skin. These are all going to be kind of multi-unit smooth muscle fibers. They're all basically independent from one another. Versus the unitary smooth muscle, these are going to be more kind of coordinated. We call these syncytial or visceral smooth muscle. And when I say syncytial, what does that mean to you guys? All working together, right? So they kind of work together to form kind of one cohesive unit. There's a lot of gap junctions between cells that allow for things like sodium to flow, so action potentials can transmit appropriately. Then you'll find they're either going to be arranged in basically bundles or sheets that allow for constriction around a specific area. So this could be the vascular endothelium, or it could be the bladder or the stomach, or whatever it happens to be. But these again are going to be under neuronal control, as we saw previously, especially with the GI tract and whatnot, but also under a lot of hormonal control. So again, we can look to see like, okay, maybe this is under sympathetic and parasympathetic activation, but also could be affected by certain hormones like ADH or renin or other things like that, or angiotensin II, I should say. So you'll find that when you're looking at the smooth muscles here that they are going to also contain actin and myosin, but they don't really have that same kind of striated appearance, right? Which is why we call it kind of unstriated or smooth muscle. But you're not going to see the normal kind of associations, like the same banding kind of pattern you'd see in the skeletal muscle. Notice there's also going to be no troponin complex here. Okay, so no troponin. So you're going to say, well, what's preventing activation of those active sites if you don't need to really have calcium activating that? We'll see where calcium plays a really big role here, affecting this as well. Anyway, still relies on ATP as well. So again, these smooth muscles will not contract unless there's ATP around. And again, no striation. So basically, instead of the Z discs that hold the actin filaments, you're going to have these dense bodies. And again, that's how I like my bodies, dense, right? And then, I'm just kidding. And so again, this is going to attach to the actin, and again, is going to be pulled by the myosin filaments. And what's kind of interesting here is that you can find that the smooth muscle cells, they can tend to contract up to like 80% of their original size, as opposed to skeletal muscle, which maybe can only contract by about 30%. And so that's because the myosin filaments tend to have these side polar cross hinges, or cross bridges, where they can kind of pull in multiple directions at once, rather than the skeletal muscle kind of only pulling in kind of one plane of action there. So again, just kind of interesting that they can contract even more than the skeletal muscle. Basically, when we have smooth muscle contraction, we want to have smooth, prolonged, tonic contractions, right? So this could be for hours, could be for days. We don't want this to be kind of we want this to be a marathon, not a sprint, right? And so we do this by having the slow cycling of the myosin cross bridges, right? We want this process to not occur very often and happen very slowly when it does occur. Basically, you're going to find that cycle of actin binding to myosin, releasing and reattaching is much slower than we see with the skeletal muscle. One of the ways this occurs is because there's way less ATPase action, and this helps to slow that cycle, right? Because ATPase was important for the myosin because it would help to break down the ATP into ADP and phosphate, and that's what got the myosin head ready for another contraction again, right? So we have way less action there, so it's going to slow everything down. But also the fraction of time that actin is bound to myosin is greatly increased. So when it binds, it tends to stay bound there for a lot longer, which allows for kind of more sustained contractions. And it also utilizes way less energy than your skeletal muscle. So about 1/300th the amount of energy needed to maintain contraction of smooth muscle versus skeletal muscle. Because again, we're using a lot less ATP in general. So how does this occur? So instead of troponin, we have a molecule called calmodulin. And so calmodulin, as you might imagine, is going to bind to calcium. And this can either come from the extracellular fluid, which this tends to be a much more important pathway for causing smooth muscle contraction. You're going to find that the sarcoplasmic reticulum for the smooth muscle is much less developed. You can kind of see a picture here where they don't really have those T tubules like we saw in skeletal muscle. They have these little calveoli, which are kind of wimpy. Indentations in the cell. They don't really have well-developed sarcoplasmic reticulum. You still get some calcium from there, but primarily it's going to be from the outside, right? But calcium is going to bind to calmodulin. You can see calmodulin here. They bind up together, and so this will activate an enzyme called myosin light chain kinase. It's a MLCK. That's myosin light chain kinase. And so this will activate that, and what that's going to do, as you might imagine, is it's going to phosphorylate the light chain of myosin. Okay. At that point, once they're phosphorylated, this will allow for the cross bridge to form. It's going to bind to actin, and at this point, the contraction mechanism is the same as skeletal muscle. Okay, so we need ATP there, be broken down to ADP and phosphate. Once they bind, still going to cause that same kind of ratcheting mechanism we saw with skeletal muscle. It's just instead of relying on troponin to uncover active sites, it's going to rely on this phosphorylation of the myosin light chains. Okay. And then in order to cause smooth muscle relaxation, basically you're going to have calcium pumps. Again, these are requiring ATP to be useful, either back out of the cell or into the sarcoplasmic reticulum. Again, this is a very slow process as well. So again, as more calcium is around, you're going to have more activation of smooth muscle. It's slower to get rid of the calcium, so it likes to stick around for longer. So you tend to have more kind of prolonged contractions there. At this point, once the calcium levels go down, you're going to find another enzyme called myosin phosphatase. You see myosin phosphatase here. As you might imagine, it's going to enzymatically remove phosphate from those light chains. And then at that point, basically it's going to restart the cycle all over again. And so what's thought to be why these smooth muscles can contract for so long is called this latch phenomenon they talk about, where once it's bound, it likes to stay bound. And the reason for that is because basically you find that you have a lot basically kind of low myosin phosphatase activity around. So if you kind of depress the activity of that enzyme, especially by keeping calcium around in a high amount, that allows for that once the bind to occur, it just stays there and hangs out until you have basically the phosphates come along and unbind that phosphate there. So again, allows it to bind for longer periods of time, less energy being used, so it's a very efficient kind of process. So looking at nervous and hormonal control, we've already covered a lot of this before, so I'm not going to kind of belabor the point here so much. But certainly we have autonomic control, having acetylcholine released onto things like cholinergic receptors. Notice here that this is not really nicotinic mediated anymore. This is going to be more cholinergic through the parasympathetic nervous system. And then on the flip side of that, you'll have the adrenergic receptors being activated by norepinephrine primarily. So again, keep in mind the different tissues will respond differently. So for instance, if I have, say, activation of acetylcholine receptors in the bladder, what can occur? Acetylcholine affecting the bladder, if those muscarinic receptors causes contraction, right? That's when urination. Versus the adrenergic receptors typically cause relaxation. And on the flip side of that, if you're looking at, say, like the vascular endothelium, causing activation of alpha receptors causes what? So alpha activation of the blood vessels causes them to, by norepinephrine, constrict down, right? Because norepinephrine, it's a vasopressor. It increases contraction of the vascular smooth muscle. You cause an increase in blood pressure. Muscarinic effect's not really so high there, but just know that different tissues are going to have different effects. We've covered a lot of this already. Previously, I can tell you guys are already well versed on all this, right? That's okay. It's cobwebs. It'll come back to you guys eventually. Okay, and then looking at action potentials, obviously calcium is much more important. You'll find that instead of relying on sodium channels to propagate those action potentials, much more focused on calcium. And again, those calcium channels are much more slow to open and also close, which is again another thing that favors long-term contraction of the smooth muscle versus the skeletal muscle. Okay, again, you guys remember things like the slow waves? You guys remember seeing where we saw these at? Remember the GI tract, I'd have that kind of slow wave, which would kind of undulate, and once you got to a certain point, you'd be more likely to have action potentials, right? The resting membrane potential kind of flow. Places like the uterus, you would end up seeing kind of plateau phases here. We kind of have long, prolonged contractions. So you can just know that the action potentials are a little different. Mainly going to be mediated through calcium, though, not sodium. So again, you can find that, especially with hormonal control, about 50% of smooth muscle contraction is initiated without any kind of action potential affecting it, which is good. And you guys remember back on the autonomic control, how things like acetylcholine would affect potassium channels, which affected the resting membrane potential. So in some cases, you're going to find that these are going to be affecting the resting membrane state rather than specifically causing activation of contraction. On this side, though, when we're looking at more hormonal control, certainly you can see local tissue factors being a role here. So think about things like blood vessel dilation happening in response to a lack of oxygen or excess CO2 or decreased pH, right? All those are things saying, hey, we need more oxygen to the tissues. Let's go ahead and dilate so we can get more blood flow happening here. And then certainly hormones, we know things like epinephrine from the adrenal gland, angiotensin II, vasopressin, again, we've covered these ad nauseum. Certainly things like histamine. You guys know what histamine does to blood vessels? Should dilate, right? That's why you get hypotensive when you have an allergic reaction. Some of them will cause ion channels to cause, open ion channels to cause activation. Some of them will only just affect resting membrane potential, but we kind of covered a lot of these already. So just realize that, the smooth muscle, much more under control of both the nervous system and hormonal control, versus skeletal muscle is really all just the nervous system affecting that. Okay, that covers it for muscles. Everyone feel fatigued now? Go eat your muscle milk. Milk your muscles for all they're worth. Any questions? All right. If not, I will see you guys next time.